second of our fall rigor and reproducibility workshops. As always, we're delighted to host some exceptionally engaging speakers who are going to give you a glimpse into the challenges of rigor and reproducibility, as well as some of the repercussions of non-rigorous science, particularly in healthcare. Though we will just scrape the surface of some of these issues, we hope that you leave here with, a better understanding of what these challenges are and have some ideas in your own research, in your own labs, for how to better address these issues. I will note that this is our introductory workshop, and we do plan to host our more in-depth summer workshops. One of those is on computing in R. We have one on publication and reporting, and then we have a new one that's focused on imaging. So be sure and stay tuned to the GCC News. Again, those are typically offered during the summer. So first off this morning, we'll hear from Lee Ellis, who's getting his slides ready, and Keith Baggerly after that. And I'll turn it over to them in a minute. But again, just want to cover a few things. You'll notice on your agendas that you should have received in your email that after Lee and Keith speak, we'll take a break from 11:20 to 12. And this is for those of you who may be working from home, taking care of kids. We want to give everybody a break to just do what you need to do, to also grab a lunch and return to your computers so you can participate in the experimental design panel. So for that experimental design panel, you were asked to submit questions in advance, and we've selected a few of those, and we'll be discussing those with our panel. We will have, as part of that, we have some audience response polls, and then several different areas of expertise that are represented in our, among our panelists. But we also know that there's a lot of expertise among you as well. So we hope that you'll be engaged in the chat. If you don't feel comfortable unmuting yourselves and asking questions, we'll try that. If you'd like to unmute and ask a question, we've got quite a few on the in the workshop today, but we can see if that'll work. But if you don't feel comfortable doing that, please feel free to just enter your questions in the chat. Otherwise, I would ask that you do please make sure that your microphone is muted. One other thing is for our afternoon sessions, we are going to have breakout sessions. And so here in the next few minutes, you're going to receive an email from Dawn, who is going to tell you what number your breakout group is. And once you enter the breakouts, those happen for our data management and publication and reporting sessions. You'll be prompted to enter a breakout room. And once you do that, you'll need to quickly designate a person to be a group leader that will report out when you come back. Each of you will be reporting out at least once between the two sessions, and Melissa and Stacy will call on those groups randomly. So you want one person that's going to be the person that reports out, and another person that is going to record the ideas that came out of this group. And we ask that the recorder email those notes and distinguish them clearly between the data management breakout and the publication and reporting breakout, and then email these either to Dawn or myself. I think that pretty much takes care of everything. I will note that in your upper right-hand corner, when you're in the breakout room, you will have a timer, so you'll know exactly how much time you have. And other than that, I think that about covers it. Again, we're really happy that you're here, and I do want to take a moment to just thank our speakers and our panelists who do this over and over again, because they are so committed to this cause, and I truly believe that it's making a difference. So thank you very much, Lee, Keith, Stacy, Robia, Melissa, all of you, Kurt, and Natasha. We really, really, really appreciate it. We couldn't do it without you. So with that, I'll turn it over to Lee.
Great. Thank you, Suzanne, for your efforts in keeping this going, even through the pandemic. This is fun. We do this a lot. I want to let you know that unfortunately, I have to continuously update my talk, because there are so many crazy things that go on in the world of research integrity that it warrants updates, including just about an hour ago. So on my cover slide, I list some of the roles that or positions that I hold. Of course, I work at MD Anderson, have a research lab. I work in SWOG, which is a NCI-sponsored clinical trial group, but probably where I encounter or most aware of research integrity is in my role as a reviewer and a deputy editor of JAMA Oncology. And previously, I was on the editorial boards of Clinical Cancer Research as a senior editor and Cancer Research. So that's where I think more of you would submit papers rather than JAMA Oncology, which is primarily clinical, but you learn a lot by being in these positions and working with other editors. So let me try to advance the slides. Here we go. So what we're going to talk about today is, frankly, the issue at hand and the spectrum of research integrity or lack thereof. Why does this occur, and how do we fix this, or how do you address this? And if I had more time, which I don't have, I used to play a game of Find the Fraud. There'll be a little bit of that as I show some work from Elizabeth Bick, but perhaps some other time we can do a Find the Fraud drill. So if we used an audience response system, which we're not going to be doing because I can't even get, we're not even able to see you raise your hands. I'm sorry, we can't do this in person because it's really fun to see the expressions on your faces. But at the end of my talk, you will feel entertained, I hope. Anytime you give a talk, that's a goal. You will feel angry. Some will. I'm sorry, I won't be able to see your faces, as I mentioned. You will feel discouraged. I don't want you to feel this way, but somehow for the next 24 hours, you may need to drink a bit more. You may feel reinvigorated that it's okay not to publish in Cell, Nature, or Science, and you may have a beer celebrating that. And you're likely to feel a little bit of all of the above. So everything you need to know about research integrity in one slide. And ahead of time, I've asked Suzanne and Dawn to provide some materials for you. So on the next slide, as you will note on the right-hand side for research trainees, it's what you need to know about research conduct. And this was really a nice summary put out by the Office of Research Integrity, which is part of Health and Human Services of the government, of course. And just keep this on your desktop in case you ever need to refer to this. But this is really a good resource and only came out about 3 months ago. And then there's retractionwatch.com, which I will talk about in just a minute. And you can see some of the titles that come out daily in an email describing where people have probably not done the right thing. So this is kind of an overview of what you see from retractionwatch.com. I will say I have no financial or political interest in this. This is run by Ivan Aransky, who's a professor at NYU, and he and his colleagues just couldn't stand it anymore. So they put together this daily email as well as a weekly summary of everything. And it's quite interesting. And I actually met him at an FDA meeting. So he's the real deal, and he's not out to make money by advertising or anything like that. But you can see some of the titles for some of these retractions, such as Science Retracts papers, a paper as authors blame the pandemic for the issue of image manipulation. Of course, we've all got COVID fatigue, but that doesn't allow us to falsify data, at least not yet anyway. And this came out this morning. And I kind of found this fun. A journal retracted a paper published earlier this year for pretty much every sin under the sun, scoring an own goal in the process. This was an interesting read. I'll let you look this up yourself. Just go to the website, retractionwatch.com, but some very interesting stories. And the people who write this have personalities, which is a nice change of pace in this crazy era. Now, this doesn't even cover it all. We had a workshop, as Suzanne mentioned. We do this workshop often. I summarized the articles that had already been retracted related to COVID publications a couple months ago. There were about 31 to 33 retractions. And I checked this just a couple days ago, or maybe it was yesterday. I can't keep track of time anymore. And there have been over 40 retractions of papers on COVID. So you see the papers come out, you see the headlines in every newspaper, whichever way you stand on masking or social distancing or hydroxychloroquine. You can find the paper that supports your point of view, but go back later on to see if it was retracted or not. And I spent a lot of time on retractionwatch.com because it's so interesting, but I also want to mention PubPeer. Now, PubPeer is a publicly available source, pubpeer.com, and you can go to it, and here's where people can comment on other people's papers where they find some issues. And this is just one example. You can leave your comments anonymously, which may or may not be a good thing. But here's an example of somebody who noted a figure in a paper and noted that there were lots of duplications. And the way that people now report duplications is that they use a box. So anywhere you see a yellow box here, it's a duplication. And once you take a look at this, you can see that it's relatively straightforward and easy, but without somebody leading the way by showing these red, blue, green boxes, whatever it is, you may not recognize this in a paper. And that leads me to the next slide, or that was going to lead me to the next slide. I thought I had this. I do have it on the following slide. Sorry. As you can tell, I change my talks every single time. So there's a difference between retractionwatch.com, where the editors comment on retracted papers, whereas in PubPeer, these comments can be anonymous, and it could be the arch enemy or ex-spouse or whoever may be writing some of these comments. And you really need to look at PubPeer carefully. But oftentimes there may be some arguments on PubPeer. But when you see what I showed you on the prior slide, where there's image duplication, I don't think there's much question there. Now, journal editors don't review PubPeer as a policy, but it is reasonable for somebody who has a concern to write to the editor and say, as posted on PubPeer, and here let me summarize the results, there appears to be a problem with the paper that was published in your journal. So there's all kinds of ways to utilize these resources, sometimes evil, sometimes good. This really isn't a picture of Elizabeth Bick, but this is her own posted picture on Twitter. I do encourage you to follow her if you're a Twitter fan. I'm not an Instagram or Facebook person, but I assume she likewise has a site there. And Elizabeth Bick was a postdoc with two heroes in the field of data reproducibility, Ferric Fang and Arturo Casadevall, also people that you should follow. And Elizabeth has a photographic memory where she can remember not only gels from earlier in a paper from figure 1 to figure 6, but she can remember gels from 5 years ago, which is incredible. And here, this just comes from her Twitter feed. It was a little hard to cut, copy, paste, and all that kind of stuff into a slide. So I just took this from her Twitter background. But you can see, as I mentioned, the elliptical boxes here, if you will, show areas of duplication where somebody very nicely on Adobe Photoshop was able to create the gels that they really wanted to have in reality. And she brings up some really interesting things. She doesn't have an axe to grind that I know of, and lots of what she talks about is not in the field of microbiology. It's in the field of biology in general. So a nice Twitter feed there. And she actually systematically looked at this, and I mentioned my colleagues and friends, Ferric Fang and Arturo, on here. And this looks at the percent of papers with potential inappropriate image duplication, and you can see that it ranges anywhere from 2 to 6% across the board. So if you open up a journal, there's a good chance that one of those papers will have some image manipulation. Whose fault is that? I always think it's the responsibility of the principal investigator to make sure that everything that comes out of their lab is real. I had lab meeting Wednesday of this week, and I questioned things from my lab just to make sure that the actin bands, which were tilting in one direction, and the bands for the protein of interest were um, straight across. I always want to make sure that anything that comes out of my lab is real, and then the people in my lab who are much smarter than myself explain it to me, and then I say, "Oh, okay." But here she looks at the number of papers or the percent of papers that may have some image duplication or manipulation, and you need to be aware of this. And I just can't help but update things. As I was scanning through some of the journals where I get the e-table of contents this week, you will note that there's a few authors here who seem to have a habit of doing things that aren't particularly respectable. And you can see a number of retractions that have occurred over a number of years, but it seems to be a group working together where there's all kinds of manipulation of data or unfortunately misconduct in some cases. So this issue is real. Okay, everybody take a deep breath, including me. I'm going to try to move on to some things which are a little bit more upbeat. So I've tried to update this slide every time I give the talk, but this slide is probably as good as it gets because being a cancer doc, cancer drug development, we fail way too many times when a drug enters preclinical studies, but in particular phase 1 studies. The percent of drugs that get approved are pretty low, on the order of 3.5 to 5%, and that's not enough. However, I will say that as we learn a little bit more about the molecular drivers of cancer, I think over the next couple of years, our success rate will go up. And I'm happy to report one of my friends and colleagues at MD Anderson has had a 5th drug approved based on some phase 1 studies he has done with molecular profiling of tumors and then finding the right drug to hit that driver mutation. So I think we're going to do better, but still across the board, it's about a 5% success rate for cancer drugs going from phase 1 to actual FDA approval. And I go back to my friend Bob Rudinsky, who no longer looks this young anymore, but it's been 20 some years since we took this picture. And he moved to Amgen soon after the year 2000. And as I was trying to reproduce a bunch of studies preclinically, mostly in GI malignancies, I called Bob, and we were just talking, and I said, Bob, most of what I'm doing, I can't reproduce. And he said, Lee, do you realize that most of what's published in academia cannot be reproduced? And he really stressed the word academia. Now his boss and colleague at the time, a medical oncologist and scientist, Glenn Begley, had been prospectively collecting data from studies that were done at Amgen, and not just prospectively collecting the data where Amgen was trying to reproduce studies in academia, but he would actually meet with the academics over breakfast or phone calls or emails to try to figure out why he couldn't reproduce, or the Amgen team couldn't reproduce what the original authors could produce. And he never really got an answer. I'll show you the data for that later on. I can tell you one anecdote. And one time he met somebody over breakfast, and they said he couldn't reproduce figure 5, and they talked for a bit, and the PI said, well, we did the study 6 times, and on the sixth time we did this study, it worked, and that's what we reported. The other 5 times it didn't work. So that gives you an idea of the need to publish positive data and have positive results all the time. Way back when, in the 1990s, we thought we had a cure for cancer. You can see these are called magazines. I don't think we have them anymore, but uh, there was a tremendous amount of enthusiasm over a drug discovered up in Boston. And people really thought we had the cure for cancer. And none of this panned out. And there were actually 3 drugs and then 2 more by another colleague. And none of these drugs panned out. And it turns out that with one of the investigators who was leading the first 2 drugs that appeared to cure cancer, he would not allow anybody to look at his mice in the animal room. So my principle in the laboratory is when we go sacrifice mice, I have 3 people down there, one of which is the lead investigator, but 2 of which are blinded to the groups so that there's no bias when they assess tumor growth. Now, this summarizes, published reports on issues with data reproducibility, and at the bottom is the study published by myself and Glenn Begley from Amgen, where only 6 out of 53 studies could be reproduced by the company. And you'll see a lot of these come from companies because they're trying to take what we do in academia and translate this into benefit for our patients. And likewise, Bayer had a very low success rate, but there seems to be a really poor reproducibility rate in neurologic disease. And when I did have the chance to meet with leaders of the NIH divisions with Francis Collins and a number of other people on campus, the folks from the neurologic branch of the National Institute were really paying attention to what Glenn and I had to say because they've had a hard time reproducing studies. And you can see at the very top, 0 studies were reproduced from 70 drugs and 221 independent studies. So this was a study that Glenn and I published. It did raise awareness. We did have some back and forth with Nature, but eventually we published what we wanted to say, whereas sometimes Nature has some of their own thoughts on what should be put in the paper, but we stuck by our guns. Being in Texas, I can say that. And this was published, and it did get quite a bit of attention. Following this, you had a number of people who jumped on this issue. I'm not saying jumping on the bandwagon, but who are also interested in this issue. And they looked in various fields. And as I mentioned, there was quite a bit of lack of reproducibility in published studies. And this was collated by Len Freeman in PLOS Biology in 2015. And Len has done some more work in this area. Nature then, after giving us a hard time about publishing this work, then jumped on all over this, and they did a survey in 2016 asking readers what they thought was there really a reproducibility crisis. And as you can see here, anything in red or a lighter color of red, pink, if you will, pink or orange, a lot of people think there's a lot of problems with reproducibility. And most of that would come from postdocs who are actually in the lab trying to prove or disprove a hypothesis. But I give credit for Nature turning around and really asking people, what do they think about this? So I'll go back to my own slides here. This is, I don't think everybody doing research and has data that can't be reproduced, I don't think they're all evil people. I think when you do research, there's a full spectrum that we cover. Some people are honest and will report negative studies, although journals don't necessarily not like negative studies. Some people are sloppy. We'll talk a little bit about cell line identification and things of that sort. Some people are only report positive findings, which is not truly honest, because you should always report the denominator as well. Sometimes there's a lot of important information in negative studies. And then there are people who falsify data. And then at the very end of the spectrum, there are people who fabricate data. And sadly, I think we're moving in this direction. And I've used this slide for a number of years, and I'd like to be able to say that things have remained stable. But as I just showed you from experimental cell research this past week, there's lots of retractions out there, and it means that we're really not stabilizing things or moving more toward the left, but we're kind of moving toward the right again. So there are consequences of this. It's not just about a postdoc or a graduate student's career or PI's career. If we, as I mentioned, with the endostatin studies that made the cover of several magazines, clinical trials are bound to fail if we are not honest in preclinical studies. There's a tremendous amount of wasted time and effort of investigators and trainees. There's a tremendous waste of money, as I'll bring up in just a moment. And there's a loss of confidence from our community. I mean, we ask our community to donate to the American Cancer Society or our cancer center, or whatever it may be. And then when we see that all these studies with great promise fail, or we see an improvement in survival of 1% or so, I just don't think that that hits the that's a goal for the community who are donating their money, especially in this time when there's such a need to support so many causes. That why would they donate to cancer research when people are starving on the streets right now? You have to pick and choose, and I actually had to do a bit of that yesterday. As I donate to the Houston Food Bank, but also try to support some cancer organizations and dog rescues. Don't forget dog rescues too. But you know, you've got to have confidence in your community to raise money to do cancer research. Going back to Len Friedman, he looked at the economics of reproducibility in preclinical research, and you can see that once you drill down, because of the amount of research that is done that is not reproducible, we are wasting about $30 billion a year in the US on irreproducible research. $30 billion. I don't see any head shaking, but I don't see anybody, so that's why. All right, let's move to something that's probably occurs in a lot of labs. Doesn't mean they're bad people. It just means that it leads to research that can't be reproduced. Keith will talk about inappropriate statistics. I'm not going to spend a lot of time there. I will spend a little time in a survey later on talking about cell line contamination and drift. But I want to talk a little bit about the fact that journals don't like negative data. I mean, there is a journal called the negative studies or whatever it may be, but this is not what you are seeking when you start a postdoc or start graduate school. You're not seeking to have negative data and say, look, I have negative data and it's really negative. I want to get this published somewhere. That's not an easy thing to do. So since journals don't like negative data, principal investigators don't like negative data. We have careers too. We have faculty evaluations. We get promoted or not promoted. So all these issues are real for us too. So as I mentioned, journals prioritize positive studies. And if a drug works in 2 cell lines and doesn't work in 8, and that may be important, that may tell us something about a negative biomarker. We only see the results of the 2 cell lines where it worked, whereas probably, as I mentioned, the most value may be in the reasons why it didn't work in the other 8 cell lines. Students, postdocs, and faculty need publications for advancement. It's the old adage of publish or perish is true. And in many labs, 2 trainees work on the same project, competing with one another. Oftentimes this happens at a city up in the Northeast. Gets cold about this time of year. And when you have 2 trainees working on a project, the one with the negative data probably doesn't look good to the PI in that laboratory, because he or she is not helping that PI with their goals to move up the ladder. So we tend to only report positive data and we ignore the negative data, whereas I keep saying there's probably a great deal of value in the negative data. So people have written about this. We need to highlight negative results to improve science. But the bottom line is, this is where I put my editor's hat on, I personally will always consider a publication where there's a negative clinical trial. It didn't meet its primary endpoint. as long as the authors try to explain why we didn't hit the primary endpoint and why the trial was negative, and hopefully there's some biomarkers in there. So I may be the outlier as an editor, but I think there's a lot of information and knowledge gained by reporting negative trials, but there are more people who don't feel that way. So you're not going to see a lot of negative trials in the big journals, but this is important, especially the negative clinical trials. These should be in New England Journal of Medicine, JAMA, Lancet, etc. All right, this is the hard part. Let's talk about falsification and fabrication. This is misconduct. Now, do investigators intentionally falsify or fabricate data? Because none of us, and the 67 people, or how many people are here today, I can't even see, but in the large number of people here today, none of us would ever consider this. And after Glenn and I published that paper in Nature, I received about 90 emails that looked like this, and some of them were so caustic that I had to black it out, even for this esteemed group in the audience. So at the top, you can see that here's a PI who had to change his lab 7 times because he would not give the principal investigator the data that they wanted. And the PI wanted the investigator to falsify data, and they refused to do so, and they kept having to move to a different lab. Now, I have to give this person credit because finding 7 different jobs in 7 years is a hard thing to do, especially when you don't have a letter of recommendation coming from the prior, your prior boss. Does fraud really occur? Well, here was a paper in Nature in January 2014, came out of Japan. And it turned out that there were all kinds of flaws. And in investigating the research, the committee found that the first author had manipulated data in an intentionally misleading fashion, and they branded it scientific misconduct, and the paper was retracted, withdrawn, whatever you want to call it. However, more came out of this, and sadly, the head of the institute where this research was done committed suicide because ultimately the boss of any institute is responsible for what goes on within that institute. And it was very sad to see this come up only 6 months after the publication of the article itself. So this was a quick retraction, and sadly, the death of a director of an institute. Okay, does it occur in the clinic? Well, Keith Baggerly gives an incredible talk on this. I don't want to steal his thunder because I can't. His talk is entertaining and funny, but you will hear a tremendous amount on the Anil Patti story, which is just still shocks me that people didn't jump on this earlier and really recognize all the badness that was going on at Duke, even was not recognized by Duke itself. And Keith will talk about this, and you will really enjoy this talk. Unfortunately, there are more issues in clinical trials, and the Chinese government actually admitted to the fact that 80% of the clinical trials that were reported out of China were fabricated. Falsified is one thing, fabricated is completely made up. So I will just leave it at that. A couple years old. I haven't seen anything since that time, but I think this is one slide that I keep with me. So what's the impact of retracted clinical papers on patients? And you can see the data in these tables here that came out of the Journal of Medical Ethics now about a decade ago. But for each retracted paper, you probably impacted about 2,600 patients per paper. And when the papers were retracted for fraud, 1,500 patients were impacted. So this is just not about careers, but when people, let's say, as I mentioned with the endostatin story, they take on something which is not reproducible, and then they begin a clinical trial and it doesn't do anything for the patients, and which meaning that it actually did harm because you put a patient on a trial for 4 to 6 months when they were actually getting something which did nothing for them while they could have been on a different trial, possibly benefiting. That's not a good thing to do. So this preclinical studies do have an impact on our patients, because our ultimate goal is to move stuff from the laboratory to the clinic to help our patients who are in need and who trust us. And I updated this for the last talk, and I think it's very relevant right now. These are three, what I consider major fraudulent papers, series of papers from Macarena from the Karolinska, which came out over a number of years, a surgeon who claimed to be able to create tracheal implants from stem cells. And there was a breast cancer one that you can see at the top, breast cancer and bone marrow transplants, which was completely falsified. But the one we really need to pay attention to today is the one from Wakefield in 1998 in the Lancet, connecting autism and vaccines. And obviously this has implications for why 40% of people today may not want to take the COVID vaccine when it becomes available to them. And when I did a deep dive on this, I was shocked, and I thought I knew the field fairly well. So I'm not going to read through this slide. You can read through it quicker than I can read it to you. But I did highlight one item below that even before the publication, Wakefield was working on a competitor to the measles vaccine, which he would profit from. And you'll see there's a lot of financial conflict of interest in Wakefield's paper in the Lancet, which was completely unethical, unjustified, was not backed up by any data whatsoever. And some people believe he didn't even, conduct a study at all. So this paper was retracted, but to this day, people still think vaccines are bad, and it's mostly based on this paper. So now you know. No institute is immune. MD Anderson has had its own issues. Of course, we're a large institution now, 22,000 people, but we've had retractions and people who have found guilty of misconduct. And then there's Harvard. Harvard has had its issues with misconduct, again, just like any other institute. And then there are people who are outstanding leaders in the field. Bob Weinberg would be considered the leader in the world in cancer biology, trained a number of outstanding people, including friends of mine and mentees of mine here at MD Anderson. And Bob has had a number of retractions, but remains unscathed by it. And there are lots of comments after some of these retractions on various websites. And this does kind of point to the fact that when you go into a lab such as Bob Weinberg's, you're really pressured to publish in CNS, Cell, Nature, and Science. And this allows you to cherry pick the data or lead you to cherry pick the data, as I mentioned before, selective reporting. And you can read the rest of it, but it doesn't matter where a paper comes from or who the author is on a paper. Read it carefully, look at the blots, see if you can become an Elizabeth Bick and figure out if there's any manipulation there, and then see if you can reproduce the study yourself if you're interested in that field using the methods that they had published. And do not hesitate to contact the PI if you can't reproduce results. So as principal investigators, we can't have large labs with 30 people running around and only meet with them once a year when they have data for a Cell, Nature, or Science paper. What we need to do is we need to keep track of the data in real time. And I can say myself, I have my weekly lab meeting. Even during the pandemic, we review every single blot. And as I said, I even mentioned something on Wednesday about some bands which didn't look good. So if you're a PI of a laboratory, you're responsible for absolutely everything that comes out of that laboratory. So keep track of that. Right after this paper came out, Len Zwelling pushed several of us to do a survey at MD Anderson, which we did. We included 2 postdocs as co-authors. Aaron and Russell were postdocs at the time, and Suzanne helped with the data. And of course, Len and I oversaw the study. And we had, once the study went out through MD Anderson, it was an IRB approved study, we had 240 responses to the survey in 6 hours, which we found remarkable at a time when, people tend to have other things to do. And we quickly had 311 responses in 3 days. So we asked, have you ever tried to reproduce a finding from a paper but not been able to do so? And nearly everybody, or not nearly everybody, but about half of the folks, no matter who you talk to, whether it's senior faculty, junior faculty, or trainees, they've all had issues with reproducibility, which, is not too far off from what we have found in other studies that I've talked about already. It's, approaching 50% in junior faculty, and the older you are, the more likely you are to have run across difficulties in reproducibility because your career is longer. Almost 70% of senior faculty members couldn't reproduce something that was published in a paper. What were the driving forces? Well, were you ever pressured to publish findings of which you had doubt? 22% of people were pressured to publish findings, even though you weren't confident in what your outcomes were. Have you noted pressure from a mentor to prove that his or her hypothesis was correct, even though the data you generated did not support that? And that's about a third. I mean, that's a lot. Every time I give this talk, I still think, wow, that's a lot. Are you aware of mentors who have a high impact publication, who require a big time paper before a trainee can leave the lab? And half the people know a mentor who won't allow you to finish your postdoc or finish your graduate studies until you have a CNS paper. So that's a problem. Now, again, sometimes these stories come out in the comments that are left. It came out from the Nature paper, and here it came from the survey. And one thing I didn't think about, I thought I had a good handle on this by then, but one thing I didn't really think about is that international trainees need to show productivity to stay in the country. And I'm staying out of political issues right now, but this came from the paper, nearly 7-8 years ago now, where the PI pushed them to give them the data that they want, and in order for them to stay in the country and to keep their position, they have to publish. They have to stay in the PI's lab and continue to get paid and continue to have a position, and they have to do what the PI want. And the rest of this you can read on your own. We then followed this up with a survey on reproducibility. I think, Keith, if you ever want to make sure that your paper is 100% correct, include a statistician on the paper. So we included Keith for his insights, and also to make sure that we did our stats correctly and not conveniently. So we published this a couple of years ago in Clinical Cancer Research. And we had a population of 467 people who returned this survey to us, and you can see this occurred in all types of fields, but most of the people who completed the survey were postdocs. And here I've tried to highlight the important issues here rather than going through each one, but how often do you authenticate your cell lines? And about 40% of people never authenticate their cell lines. In my laboratory, we do it around Christmas. We just did it because it's easy to remember. Oh, it's Christmas. We need to do our cell lines. Let's give ourselves a gift. And then we do it 6 months later. So we do that in June. Also testing for mycoplasma. About 30% of people never test for mycoplasma, which is a very easy test to do. And certainly mycoplasma can alter your cell line characteristics. Do you and your lab collaborators perform blinded studies? And a third of people never perform blinded studies. I mentioned when we go back and sacrifice animals that 2 out of 3 people down there don't know which group the animal belonged to, but I would encourage or almost mandate, if I could, that people do blinded studies. That's certainly how the coronavirus vaccine was done, blinded studies. That's the right way to do it. And Keith may comment on this later on, but we all have biases, and we need to avoid those by doing blinded studies. And do you consult with a statistician regarding your studies? And half the people don't believe in statisticians because, well, they have software, so they know what to do. You keep you keep doing studies until you get a p-value of less than 0.05, and then you're done. I'm sure Keith's not very pleased with that approach, but that's the viewpoint of many of the postdocs that I've run into. Do you feel that in order to get out of the lab and get an academic position that you have to have a CNS paper, and about 50% of people, or higher, more than 50% of people feel this way? Do you feel the pressure to publish or perish influences the way you report your data? And 60% of the people believe in that. And have you ever witnessed somebody falsifying or fabricating data? Now, nobody ever admits that they did it themselves, although you may see that in panel C. But about 25 to 30% of people have actually witnessed somebody doing something that was unethical. So this really happens, and you can imagine that these estimates are probably low. Okay, why does this occur? Well, I've already mentioned the issues of the importance to publish or perish that occurs both for trainees and for mentors. Uh, in order for trainees to keep their lab, they have to make sure that they remain productive. In order to move to the next level, you need high impact publications. And basically the same thing holds for faculty. I mean, we need to get promoted. We certainly benefit from endowed chairs. We need to get grants, and our biosketch is critically important when they're sent to study section to see that somebody is productive. It's nice to walk down the hallway and have somebody congratulate you for being promoted or obtaining an endowed chair or something like that. And having an endowed chair also means that you receive some finances from that endowed chair. So there's all kinds of reasons to massage data. And this is fairly consistent with what came out of the Nature survey from 2016. And I just highlight what's on the right side of the slide. Um, why do people do things? They do selective reporting, as you can see, and that's always or oftentimes contributes to why research may be irreproducible. They feel that there's a problem with the pressure to publish. They feel that the stats are not valid. So we understand the problem with reproducibility, and we all think that there are people who are massaging data, if you will, in order to try to get this into the big journals so that they get their promotion, their chair, their next postdoc, whatever it may be. Okay, let's talk about impact factor mania. This is a term that came from my friends, Casadevall and Fang. They wrote a couple papers on this. They're microbiologists, so many people here may or may not read some of their literature, but I do remember a quote from a postdoc in the laboratory when I was interim chair of cancer biology, and I had these weekly meetings with postdocs just to see what was going on. And I ran into a postdoc after the meeting in the hallway, and she came up to me and said, "My mentor just told me you are nothing unless you publish in CNS." So think about the pressure on that trainee for that after hearing that statement. And then again, Casadevall and Fang, I've written about this in MBio, a microbiology journal, and here you can see I've highlighted some of the major issues, but I want to really highlight the issue of associating the value of research with the journal where the work was published rather than the actual content of the work itself. The mania is causing profound distortions in the way science is done that are deleterious to the overall scientific enterprise. And then people started questioning, should we eliminate the impact factor? And here we have another publication from my friends. And what they did was they looked at the correlation between the impact factor of a journal and the retraction index, which they created and wrote about. And of course, one could easily say that there's going to be more scrutiny when you look at papers in Cell and Science than when you look at some of the other papers that are lower down on the list. But the higher the impact factor, the higher the retraction index. And this was also highlighted in the New York Times. And you can read the rest of this. Another paper in PNAS, misconduct accounts for the majority of retracted scientific publications. So this misconduct thing is real, and we need to be aware of it, and we need to avoid that. Now, Nobel Prize winner said he will no longer publish in these high impact papers, and I'm sure that sounds really respectable, except for the postdocs in his laboratory, where they actually may have a paper that should be in Cell, Nature, or Science, but he won't publish in that because he doesn't believe in impact factor mania. So I don't think that was actually fair to the trainees in the laboratory, but I appreciate the fact that he took a strong stand on that, but we always have to look after our trainees. Okay, last factor, last comment on impact factor mania. There's nothing wrong with publishing in Nature. I would love for my laboratory to have our next publication come out in Nature. So strive for Nature, but don't lie or die for Nature, or don't ever compromise your ethics. All right, what can we do to fix this as I finish up? Well, I wrote a review on this in 2015 in Lancet Oncology. I'm sure you can read this and memorize this in just the next 3 seconds. But it's basically most of the things that I've already talked about as we've led up to this point. So this is available to you online. Then I updated this a couple days ago. My Adobe Acrobat is not working well, so 2020 does not look as good as the rest of the cut and paste that I did. But what's disappointing to me here is the fact that we talk about all these retractions, and I showed you a group that had 6 or 8 retractions over a 6-year period of time. Yet, to me, this is a relatively small list of people who have been found guilty of misconduct. And, even in 2019, we talk about it all the time now, but only 5 people were found guilty of misconduct by the US Office of Research Integrity. So I kind of feel like we talk about it a lot, but there's no bite when we actually find somebody who has not been working under the highest ethical standards. So what are the consequences? Well, if you're found guilty of misconduct by the ORI, and this goes along with that handout we sent to you ahead of time, your papers are retracted that were found to be conducted with misconduct. You have to have your research supervised for 3 years, which I have no idea what that means. I mean, does the chair walk into your lab meetings, or does somebody else supervise it? Do they record it? You can't serve on any NIH committees for 2 to 3 years, so you get rewarded for committing misconduct, and you can still receive NIH funding. So you've taken taxpayer money, you've committed misconduct, and you can still get money from taxpayers, even though you've published fraudulent data. I have a problem with that, as you can tell. So we have to have a punishment that fits the crime when you're found guilty of fraud. And we have to deter such behavior and not reward it by still allowing them to apply for and receive NIH or DoD funding. And I think the whole system needs an overhaul, but we don't have time to talk about that today because you need some more upbeat talks by the speakers who will come after me. So I'm going to end with this. The primary inquiry rests with your NIH-funded institution. So for instance, at MD Anderson, if somebody is accused of misconduct, either through a website or by writing for your research integrity officer, we have an inquiry that occurs. The problem is that we are policing ourselves, and there's an inherent conflict of interest if we find somebody at MD Anderson guilty of misconduct. Now, it's the right thing to do, and we have Bill Plunkett leads that charge at MD Anderson, and there's nobody more honest than Dr. Plunkett. But for all institutes, and there's an inherent conflict of interest when you are your own jury. So your institute will have a scar on it if you find somebody within your institute guilty of misconduct. So what's the mechanism for addressing misconduct? And it's institutional dependent. So you can see the list here of where you go to the department chair, division head, you can go to anybody about it, you gather information, you have an inquiry, you actually can interview folks face to face. But again, there's an inherent conflict of interest because the same place which could suffer a penalty for this is the judge and jury. So I have a problem with this, but I'm not sure I have a solution with the government being able to do these inquiries every time there's somebody accused of misconduct. So if you are really not comfortable submitting a name to your own institute, most universities or university systems like the University of Texas system do have a website for abuse, fraud, and or unethical behavior, and you can submit a comment to them online. Use somebody else's computer though. So I'm going to end with this. This was a quote from my friend Mike Fish, who used to be at MD Anderson and is also a leader at SWOG. He stated, "You've uncovered a thorny problem in academia and selfishness. In moments of weakness or at the extremes, this creates an undertow away from integrity in science and public health. And this is the single biggest limitation in our field. I am a John Lennon freak. I'll readily admit that. So I have this in my living room here at home. And this is a print that says, "Give me some truth. I'm sick and tired of hearing and reading lines by seasick, narrow-minded, short-sighted hypercritics. All I want is the truth. Just give me some truth." And if you ever go to the National Academy of Sciences, and I've had the honor of serving on a committee there, Albert Einstein was a pretty smart guy too. He said, "The right to search for truth implies also a duty. One must not conceal any part of what has been recognized to be true." And this kind of is directly in line with reporting negative data. Remember, next week is the anniversary of John Lennon's death. So everybody on December 8th should pay tribute to John Lennon and always do the right thing. So with that, I will end with required reading. And I don't know, Suzanne, if we're going to have time for Q&A or if I'm over time, but I thank everybody for their interest in this topic, and I wish you all the best.
Thank you so much, Lee. As always, it was fantastic. We do have a couple of minutes for questions. If you have a question, feel free to unmute yourself and ask it. I do see Connie finding it bizarre as a taxpayer that this is even possible.
I thank you for that. That's entertaining. It's good to have a laugh during the pandemic.
Yeah.
This is Gina Tribble at UT Health, and I follow Elizabeth Bick on Twitter, and it's unbelievable the amount of abuse that she gets from other scientists.
Well, I'm sorry to hear that. I thought you were going to say it's unbelievable how smart she is, but
I truly admire her and I love what she's doing, but she also catches a lot of harassment from people in science who think that she's on some kind of witch hunt, and which is clearly not the case. She uncovers many examples of fraud all the time, and it's this mentality that we should all somehow be covering for each other rather than trying to set better habits, I guess.
Yeah.
This is part of a career path for a lot of people, but at the end of the line here is a patient. And that's what we need to think about. At least, I know we're at the Texas Medical Center, so there's all kinds of fields, in the audience here. But for me, it's that patient sitting in front of me in my Monday morning clinic that I need to take care of, and I need to take care of that patient with the best data available to us. So I'm sorry to see that Elizabeth gets a lot of abuse there. I just always click on the images because I want to see how in the world could she figure that this is a duplication or a manipulation.
She definitely has that photographic memory. But
thank you for your comments there.
No, and I completely agree with you. When we're wasting money on falsified data or on labs that aren't really focused on the outcomes and the patient, then we don't have enough money to begin with. So that's a sin in and of its own right.
That's true.
A quick question from Kelly on how do you think it affects collaboration or the person that collaborated with someone that committed fraudulent data?
Well, if you know somebody who has committed fraud, or somebody who has a reputation for that, stay away. If you're getting a cell line, do STR phenotyping or genotyping of the cell line. You need to, I have colleagues at MD Anderson who I question, and they may be smart, they may be published in all the big journals, but I won't collaborate with them. And if I give them a cell line, I give them a cell line, but that doesn't mean I'm a co-author, because then I'm responsible for the work that's done in that paper. So to all the trainees here, don't always jump on every collaboration that comes your way. Make sure it comes from somebody who is honest and truthful and somebody who you can trust, and watch the data along the way. Don't let them just hand you a paper at the end and say you're a co-author on this, but meet with them periodically if you're collaborating, even if you're just giving them resources. Stay on top of things as it evolves.
Thanks, Lee. We do have to move on. If we can answer this one really quickly, and this will be the last one. How can we reduce the publish or perish culture within academia?
I think it starts at the top. I think it's up to your chairperson, and it's up to the president or the CEO of your institute. If they value you as a mentor and not just somebody who publishes in CNS, then publications aren't everything. It's your overall value to the institute and to our mission. So I think it starts with leadership at the top, and they set the tone.
All right. Thanks again, Lee. If anybody, has any extra questions, you can feel free to type those into the chat, and we'll try to get those to Lee. But for now, I am going to introduce Dr. Keith Baggerly as he pulls up his slides. He's been with us, as has Lee, from the beginning, and continues to shock us. No matter how many times I hear this story, I continue to just have my jaw dropped. So with that, I'm gonna turn it over to Keith.
Well, good morning. I'm Keith Baggerly. I was part of the bioinformatics group at MD Anderson for about 20 years, and I love data. So and part of the fun thing about being in bioinformatics is I get to play with it all the time. But it has also turned me into something of a reproducibility zealot. And I'm going to try to tell you over the next little while about some particular stories that have led me to this particular viewpoint. I have dealt with data that is associated with high throughput biology. And what this means to me is microarrays or sequencing or whatever. It's some type of assay that gives me thousands of measurements per patient. And we collect such data just now because we have this great hypothesis that the information we need to treat the patients better is buried in all this data, if only we could figure out how to look at it in the right way. So we've just got to distill it. We've got to find it. And that's a really cool idea, but there's a big problem with it. And the big problem is that, quite frankly, human beings do not have a good intuition about what makes sense in hundreds of dimensions. What I mean by that is, if you talk about one gene and you say, oh, well, I've noticed that if this gene is highly expressed, the patients do worse. That's actually something we can develop intuition about. That's a pattern. It involves one thing. But if I go to a PI and I say, you know what, I've looked at this set of 100 genes, here's this pattern where these 50 go up, these 50 go down, and in that case, the patients do worse. They don't understand that. But unfortunately, they think they do. And I can tell you this because it's wonderful. It's sort of like a Rorschach test for molecular biologists. You can see the story evolving right in front of your very eyes, and they'll say, yes, that makes sense because this interacts with this and interacts with this, and they can craft a story, and it's wonderful. And the problem is that on occasion, we have accidentally given people an incorrect list of genes and still had them construct stories right in front of our very eyes. So what this means is that we can't trust the intuition, but if we're going to use these high dimensional data to make clinical predictions, that presents us with a conundrum. How do we trust it? Well, what this means is that for us to trust it, we need to be able to be very detailed about the individual steps which are used to assemble these predictions because those individual steps are now the things that we can look at and understand. So that's the goal. That's what we need to do. Now, as Lee has alluded to, if you go to the literature and you read the literature and you are fascinated by some wonderful finding and you want to build on it, you may be faced with the challenge that it is not always easy to replicate the findings of that study, even given all of the data they initially used and all of the code or all the descriptions of what they say they did. Trying to reconstruct that or get those same answers again is often quite hard. And most of the time, if we don't care that much about the paper, we'll look at it and we'll say, okay, we tried, and we'll set it aside. But on occasion, the goal is really, really clinically important, and our collaborators really want to build on it. So we have to do something. We have to understand it. And what that led us to do over the years is to invent a new subdiscipline of bioinformatics, which we call forensic bioinformatics. And what this is this is the art of saying, here's the data they started with, here's the results they reported, what the hell must they have done to get from here to here? And reconstructing or reverse engineering what was done, and occasionally figuring out something about the data that we didn't expect going in. Now, I'm going to tell you a bit about the application of forensic bioinformatics in the context of a specific problem of real clinical importance, which is, can we predict which patients will respond to which chemotherapeutics? I'll tell you this is important because I know that if today a woman goes to MD Anderson and she presents with advanced stage ovarian cancer, we will give her standard frontline therapy, which is going to involve a platinum agent and a taxane, and we will offer this therapy knowing that for 80% of the patients that we give this to, it's going to help them. The tumor burden will be reduced, their lifetimes will be extended, they will experience better quality of life overall, and that's a good thing. So for 80%, we will help them. The other 20%, we will poison for 6 weeks to no good effect. But we'll do this because we don't know which 20% they are going in. If we did know, we'd do something else, and that would let us do things better. So that would be really, really cool. And that's why several years ago, a bunch of people at MD Anderson got extremely excited at a new paper that showed up in Nature Medicine coming out of Duke, where the authors claimed to have largely solved this problem. So what did they do? They said, we've figured out how to make use of data from a new high throughput assay. In this case, it was gene expression microarrays, to make predictions about likely responsiveness to different drugs. Here's how it works. They started not just with patient samples, but rather with cell line data. And in particular, they're looking at data from a specific panel of cell lines that's been maintained by the National Cancer Institute for 40 or 50 years. And this panel is known as the NCI 60. This is actually a very special panel because one of the reasons the NCI has kept it around is that the NCI uses this in part to figure out how chemotherapeutics work. Specifically, if you have a drug of interest, let's say it's docetaxel, one of the things the NCI will do is it will go in and it will test docetaxel for effectiveness against every one of these cell lines. They'll say, what dose of the drug is required to cut the growth rate of this cell line in half? And using that information, they can sort the cell lines and say, these are sensitive to this drug, these are resistant, and they can use that information. Now, what's cool about this is that this information about responsiveness of these cell lines to the various chemotherapeutics, that data is publicly available data, and it exists for every single chemotherapeutic drug we use today. Now, what this means is that if we could figure out some way to extrapolate from how cell lines respond to how patients are likely to respond, then this type of data constitutes effectively a Rosetta Stone that will tell us something about which patients will respond to what. So this looks really cool. So what they do, they said, let's start with a drug of interest. Let's say it's docetaxel. We will use the sensitivity information to sort the cell lines according to sensitivity to docetaxel, and then we will pick the most extreme cell lines, the most sensitive and the most resistant, let's say 7 at each end. Then we'll use the microarray data, the profiles of these specific cell lines. Again, this is publicly available data. And what we will do with these microarray profiles is we'll look to see which genes are most different between the sensitive lines and the resistant lines. There's about 20,000 genes. We will identify the ones that are most different using immensely sophisticated mathematics like 2-sample t-tests. And we will take the top 50 and we will say the pattern of expression in these 50 genes comprises the signature of sensitivity to docetaxel. These ones go up, these ones go down. Then they moved on to the patient samples, and they looked at patient samples, they looked at the expression of these gene expression for the patients of these 50 genes, and they say, is the pattern of expression more like a sensitive sample, in which case we should give them docetaxel, or is it more like a resistant sample, in which case we should try something else? Okay, sounds moderately straightforward. The reason they got this into Nature Medicine is they said, we've now tried to apply this procedure using publicly available data throughout, and we have managed to apply it using data associated with 7 widely used chemotherapeutics, and we're getting great predictions every single time we do. This is cool. And what this meant is that within weeks of this paper appearing, we had lots of clinical investigators at MD Anderson coming to us in bioinformatics saying, we want to do this here. We want to use this approach. We want to treat our patients better. But their approach uses math, which is scary. So we need your help. Can you help us do this? And we said, wonderful, we'd love to do this. This is something we really get excited about. And furthermore, since all the data they're using is publicly available, we should be able to do this right from the get-go. We should be able to match everything. So we said, all right, let's try to do this. And in particular, let's start by doing something we think should be easy. Specifically, let's take one of the drugs that he looked at, and let's say, for that drug, what genes do we identify as being part of the signature? In other words, do we match the genes that they identify? Now, one of the reasons we thought this would be easy is that as a supplemental report associated with this paper, there's a series of tables saying, for this drug, here are the genes we found. For this drug, here are the genes we found. And within the text of the paper itself, there are also some brief blurbs that say, these genes make sense in the context of this drug because this is how the drug works, and various bits of explanation. So we were going, okay, we understand this, we can do this. So we tracked down the data, we picked one of the drugs, in this case we said 5-fluorouracil. Okay, looks good. We identified the sensitive and resistant cell lines. We contrasted the genes. I did the 2-sample t-test myself. I'm a PhD level statistician. I can do this. And we said they identified 45. We'll take our top 45. What's the overlap? Do we agree perfectly? Do we miss 1 or 2? What's the overlap? And the overlap was 0. No agreement whatsoever. And we said that's probably bad. What's going on? Well, it turns out that when you start looking at not just 1 gene or 2 genes, but thousands or whatever, we tend not to refer to genes by name anymore. We tend to use abstract identifiers. And in this particular case, since the microarrays being used were Affymetrix gene expression arrays, the identifiers for the genes were what is known as Affymetrix probe set IDs. Now, why does this matter? Well, let's see. Let's take a look at the probe set IDs that they identified as part of their set of 45 genes that are important. Here's the first 4 that they flagged and said these are important. These are the probe set IDs of the first 4 that we identified using 2-sample t-tests. Now, if you look at their list and you go from their list to ours, anybody notice a pattern here? We have an off by 1 indexing error. How did this happen? Well, we tracked down the software that they used. We looked for the documentation, and I eventually wrote it because none existed. Now, in the process of writing the documentation, we figured out how their software worked. And their software, in order to work, requires 2 inputs. First is a gene expression matrix, genes by samples, where there is a header row saying for each sample, is this responsive or not responsive to treatment with this drug? The second input is a list of the gene names in the same order as the expression matrix, but for their software to work correctly, this list of gene names must not have a header row. So if you're working in Excel and you cut and you paste and you move this column of gene names over here and you don't remove the header row, guess what happens? You get an off by 1 indexing error. And this problem is present for all 7 drugs they looked at. Now, what does it tell you about the fact that the authors were able to find an explanation for why this set of genes made sense for this drug, even though none of the genes are actually all that different in response to that drug? They can do this because our intuition in high dimensions sucks. So that's a bit of a problem. Now, while it's a problem, we stared at this for a while and we said, actually, this problem is embarrassing. We'll give it that, but it's actually not fatal to what we really want to do, which is, can we predict response? We want to predict response, and to predict response, we're actually using the underlying matrix of numbers. The labels got added on at the last step. If the numbers still let us predict response accurately, we can still use this, and this is still really cool. So we said, okay, let's back away from the gene list, and instead let's look at the numbers. Can we get good predictions with the numbers? So how good are good predictions? Well, we said, look, we've got a bar. Specifically, we said, let's say that we can get good predictions if we get predictions at least as good as the ones they initially reported. Because, hey, that was good enough to get them published in Nature Medicine, so it's not too bad. So we said, let's take a look at some of their results and see what we have to match. So on the top here, we're seeing a plot from their paper, and this is showing their predictions of likely response in a cohort of women with breast cancer who were treated with single-agent docetaxel. And in this cohort, most of their predictions for women who were sensitive to treatment, that's the blue squares, most of their predicted scores were low, that's good. And most of their predicted scores for women who were resistant to treatment, red triangles, most of those were high, that's good. There's pretty good separation between the two cohorts. It's not perfect, there is some that are misclassified, but it's mostly right. And in particular, they said, out of these patients, we classified them accurately with respect to response status 22 out of 24 times. Not perfect, but probably pretty good, and we can build on that. Okay, really cool. But I'm cruel. And what that means is that as a statistician, I like to ask people in the audience to help me with data analysis. And in particular here, my data analysis question is this: how many blue squares are there in this plot? I'll tell you that we count 13. So we see 13 blue squares and 11 red triangles. Now, why does this matter? Well, I told you that all of their analyses were based on publicly available data. So we went back to the source paper for this data, which was a paper in The Lancet from 3 years before, where the authors of that paper spent a great deal of time talking about their 11 sensitive and 13 resistant patients, not 13 and 11. And we said, this is probably not good. So let's take a look at another drug. So here's predicting response to adriamycin. For those of you who are really overly ambitious, how many red triangles are there? For those of you who aren't quite that ambitious, I'm going to simply ask you to credit the fact that there's more red triangles than blue squares. Okay, well, we thought this looked impressive. The problem is that this data set comes from a paper that showed up in the New England Journal of Medicine a few years before, where the paper was dealing with the treatment of childhood ALL, a form of leukemia. Why is this a problem? Well, it's a problem here because as cancers go, this is one we know how to treat. Our cure rate is above 80%. Now, what does this mean? It means that if in this patient population, we have a drug to which most of the patients are resistant, we wouldn't be using it. So, and indeed, if you look at the authors of these papers, they thought most of their patients were sensitive, not resistant. So what's going on? Well, we went back and looked at the software they were using again. And it turns out, I told you that their first input was this big matrix of gene expression values with a header row saying, is it sensitive or is it resistant? Well, what they actually supplied wasn't a row with text saying sensitive or resistant. No, it was a header row with 0 or 1, but they never explicitly wrote down 0 equals sensitive, 1 equals resistant. So in some cases, going from one drug to the next, they got the labels reversed. So what does this mean if we were going to use this or try to use this clinically? This would mean that we were going to recommend administering adriamycin only to the patients who are most resistant to it. This is poor clinical practice. We do not recommend this. So overall, we have a problem. Now, as it happens, all of this stuff that I have just told you about, this was not an issue of a few minutes of figuring things out. No, this was the result of the better part of 6 months of back and forth between us and the authors of this paper, because when we couldn't figure out some of these things, our first reaction was to say, can you help us? And in particular, please help us because we're at a major cancer center. We love your paper. We want to use it here. We just want your help to get it right. But by the way, are you aware that all of your gene labels are wrong? And by the way, are you aware that some of the sensitivity labels appear to be reversed? And by the way, at the end of the day, we have to tell you that if we use the labels that we think are correct, we can't get this to work at all. Can you tell us where we're getting it wrong? And their response was that, well, obviously this interaction has ceased to be productive, so we wish to draw it to a close. And we said, okay. And we said, let's do something else. So we sent off a letter to Nature Medicine describing all of the problems that we had and saying, this is a really cool idea. We'd love to predict response, but we have concerns about whether or not they can actually do it. And we waited to see what would happen. Now, if you write to a journal and you publish a letter critiquing another paper, the authors are given the right to respond. So we waited in part to see what their response would be because we weren't sure what it would be. We weren't sure if they would say, oh, we're so sorry, we'll fix it, it'll never happen again. And well, that wasn't quite the tone of the response. The response was that unfortunately, the analyses by Doctors Baggerly and Coombs are deeply flawed. They have not used our methods. Furthermore, we have double-checked our data and all of our labels are correct, including those for adriamycin. That's the one with all the red triangles. We've posted new data to our website to show this. Furthermore, we have continued to apply these methods in other high-profile investigations with high-profile publications and editorials, et cetera. So we must be right and they must be wrong. I remember reading this very clearly. One reason I remember reading this very clearly is that I was in a hotel room that evening in Bethesda, Maryland, because I was scheduled to give a talk at the NCI the next day about this very topic. And I was busy learning right before I was to give this talk that everything I was going to be talking about was wrong. So I wasn't in a very good mood that night. But I said, well, they tell us we screwed up. Let's see, maybe if we screwed up, we can figure out something to do. So they said their labels were correct for adriamycin. They've posted data. Okay, let's look at the data. Downloaded the data. Big Excel table, tens of thousands of rows, one per gene, with 2 header rows saying, is this a cell line used for training or is it a patient sample? And is it responsive or non-responsive to treatment? That's the second header row. There's 144 columns. There's 22 columns for cell lines, that's the training data, 122 for patient samples. Great, wonderful. And we said, all right, well, if we look at the data, can we at least see something? Well, first we counted the number of responders and non-responders. That should match their number of red triangles. And we said, well, their number of non-responders, it matches the number of red triangles they had. Maybe we screwed something up. Well, let's try clustering the data. Let's see if we can separate the responders and non-responders somehow. So as a prelude to that, we said, for each pair of samples, how strongly associated is it? So we looked at the correlations between pairs, and that's what I've plotted here. Now down here, these are the cell lines, and in this quadrant, what this says, this line of red dots shows where the correlation is really high. And this line of red dots says that of the cell lines, each cell line is extremely highly correlated with itself. I believe that. That's good. Okay, that's fine. And then we went to the patient samples. And this line on the main diagonal, again, we expect that. We're willing to believe that. That's fine. But what about these red dots? That's very strange. So we went back to the data and we looked at this, these 122 columns. Well, these are nominally 122 columns of patient data, But what I can tell you is that if we just look at the numerical values, not all of those 122 columns are distinct. Some of the columns have been reused 2, 3, or even 4 times. So they don't have 122 distinct patients there. Even better, right about here is one patient whose data was used 4 times, where that one patient was labeled resistant 3 times out of 4. So we're pretty sure they don't have the labels right yet. So we sent a note about this back to Nature Medicine. And Nature Medicine said, oh, well, the authors will redo the analysis using the corrected data. There's no need to publish your objection. So a full years later, the authors did indeed publish a reanalysis using only the 95 unique samples and reported great results again. Now, this surprised us because when we counted, we'd only seen 84 unique samples, not 95, so that was a bit weird. And when we looked at the 95 unique samples that they were now using, it turns out that only 80 were distinct, 15 are present twice. Of the 15 present twice, 6 are labeled sensitive one time and resistant the other. So the correction of a correction still needs correcting, but Nature Medicine had said it was done, so we moved on. We moved on next to look at some of the other papers that they said they used to prove that this worked. And indeed, here's one of them. This showed up in the Journal of Clinical Oncology, and what they did here was they extended the approach to deal with cisplatin, which is a fairly widely used drug, and focusing on lung cancer. And they looked at cisplatin and pemetrexed. They were very excited because the earlier papers had used earlier versions of the arrays, known as U95 arrays, and those required 5 chips to cover the entire genome. But almost everybody chooses just the A chip from that one because those have the most important genes. For this one, Affymetrix had updated things, so their next version was the U133 chip, and they only required 2 chips to cover the entire genome, the A and the B. But again, the A has the genes that they really care about, so that's what they used. And when they used these arrays, and when they formed these signatures, they said, wonderful, we can make great predictions, and these predictions make sense. Because if we look at cisplatin, some of the genes involved are ERCC1, ERCC4, and DNA repair genes, and there had just been a series of New England Journal papers saying this is how cisplatin works. So cool. So we got together with the lung cancer folks, and with some work, we matched the heat maps for gene expression for these 2 drugs that they were using. For pemetrexed, they report identifying 80 genes. We can match all 80 of the genes that they report, after we account for the off by 1 indexing error that they're still making. For cisplatin, they report 45. We can match 41 after accounting for the off by 1 indexing error. Now, for those of you in the audience who are good at subtraction, that leaves 4. There are 4 genes they report that the math doesn't support. They don't come from there. They come from somewhere else. What are the 4 genes that don't come from the math? Well, the 4 that they report that don't follow from the math are probe sets for ERCC1, ERCC4, another probe set for ERCC1, another one for FANK-M, which is a DNA repair gene. These are the specific genes listed as having been found that show credibility of the signature. Now that's pretty weird, but even in this context, I'm going to tell you that these last 2 probe sets, they are special. Even here, why are they special? That's because those last 2 probe sets are not on the U133A arrays that were used. They're on the other half of the 2-chip set, which was never run. So 2 of their best genes have never been measured yet. We thought this was a neat trick. We sent a letter about this to JCO. JCO sent us a letter back saying, we're sorry, we can't publish this. We sent them a letter back saying, could you tell us why? They sent us another letter back saying, we're sorry, we can't publish this. So we moved on. We moved on next to the other paper that they cited as validation, and it's right there in the title, Validation of Gene Signatures. And this was in the Lancet Oncology, and what they did here was they extended the approach to deal not with making predictions for just one drug, but rather to dealing with combination chemotherapies. And this is important because in cancer, we tend not to treat cancers with just one agent. We tend to try to hit it several different ways in the hopes that something will work. So what were the combinations they used? Well, in this case, this was a breast cancer trial, and they looked at 2 different treatments. The first treatment was fluorouracil, epirubicin, and cyclophosphamide, otherwise known as a 3-drug combo, or FEC. The other combination was taxotere, otherwise known as docetaxel, followed by a combination of epirubicin and taxotere. That's TET, that's actually a 2-drug combo, but that's what they're using. And the contention that they make is that if we had used their algorithm to allocate patients to treatment arms instead of using randomization, which is what was done here, that we would have improved the response rate from 44% to 70%. That's a big deal. So we said, all right, that's cool. We'd love to do it. Let's get the data. So we downloaded the data and we said, if we cluster the data, do we see two clusters that correspond to the responders to one group and the responders to the other? Can we use that? So when we tried clustering the data, here's what we saw. We didn't see two clusters. We saw three. And we said, what's going on? Well, when we look at the raw data come in the nature of cell files. This is something that AFI produces. And these cell files are really cool because in addition to containing for every gene an expression value saying, here's how much of the gene it is, these cell files also have a few header rows with metadata. And one of the pieces of metadata they record is, for this sample, when was the array run? And on the right, I'm plotting all of these samples as a function of run date. This first cluster, that's down here, black circles means this is a patient who was treated with FEC, one of the two drug combos. The green X's, that means that sample was excluded from the final analysis for one reason or another. This second cluster, here's the second group of patients we treated with FEC. This third cluster, here's the patients who are treated with TET, all of them. Now what this means is that we have complete confounding of the experimental design. All of the samples from patients treated with one of the drugs, drug combinations, TET, all of those samples were profiled 6 months or several months after all of the others. That gives the opportunity for a systemic distortion that's unrelated to the biology. That's a problem. Furthermore, if we check more of the data, we see that not only were these samples run months later, they were also run in a completely different lab in a different country. So as it turns out, we're certain that some of the differences they're finding aren't related to the biology. So, grumph. Well, all right, we're not happy with the design. Is there still something we can learn here? Well, how about how they're making predictions for combinations? See, we sort of understood how they were making predictions one drug at a time. They're coming up with a number that corresponds to a probability of response. How do they combine those predictions for multiple drugs? So we actually went back to their Nature Medicine paper, where at the back of it, they say, we think we have an idea how to do this. And they outlined it for a 4-drug combo called TFAC. And they said, this is probably what we do. So we said, okay, we have sort of an idea what they're going to do. And now we have some evidence about how well it should work for a 4-drug combo, a 2-drug combo, and a 3-drug combo. So what are the rules that they are using? So let's start with this 4-drug combo. The rule that they're using is the probability that a patient will respond to being treated with this 4-drug combo is the probability they will respond to drug 1, that's T, plus the probability they will respond to drug 2, plus probability of response to drug 3, plus probability of response to drug 4, minus the product of all 4. Now, as Lee noted, I can't see everybody in the audience, and that's sort of unfortunate, because if there are any statisticians in the audience, I know that all of you should have an expression on your face that says, wait a minute, that's not right. And indeed, the authors knew that something wasn't right because all of the probabilities they got using this rule were bigger than 1. And they said, well, we know that's not what it's supposed to be. We know the smallest one should be 0, the largest one should be 1. So we'll fix those and linearly interpolate everything else down to match. This particular interpolation step is not described in the methods section. The way we figured it out is we actually tracked down the Excel file that they used. We moused over the cell and said, oh, that's what the formula is. Okay, fine. And we're not happy with it, but okay, we can sort of understand it. But that also leads us to ask, what are they using for the other drug combos? And in particular, what about this 2-drug combo? Because actually this combination formula is okay for 2 drugs. It's the combination rule for independent events. They just generalized it incorrectly. So what is the rule they actually used for 2 drugs? Here's what it is. This does give us numbers between 0 and 1, but it's different. Now that's sort of disturbing. What about the 3-drug combo? Well, here's the rule that they used. I have a standing offer to buy dinner for the first person who can explain to me where this rule comes from, because I have no clue. I have no clue what's going on here. But what I can tell you is that each rule is different. Now, to paraphrase a rather famous American, as a bioinformatician working with gene signatures, I have a dream. My dream is that if you are going to validate the performance of a gene signature, that you would keep that prediction rule the same in the validation experiment. But that didn't happen here. So, okay, fine. Well, we're not happy with the design, we're not happy with the rules. Is there anything else that we can still glean from this? Well, maybe if we just give up on the combinations and say, what if we just look at one drug at a time? Can we make predictions there? We still sort of understand what they were doing there. Well, actually, the authors thought of that, and they said, if you want to make predictions based on just one drug, well, here's our best ROC curve. And this is trying to report response in patients treated with FEC if we just look at the predictions made with Cytoxan. And for those of you who work at ROC curves, this is a beautiful ROC curve. It's almost perfect classification based upon the score that they get. Wonderful. From the raw data, we attempted to reconstruct an ROC curve of our own. This is what we got. The two curves do not look that similar. There is also a different problem, and this time it's not really a mathematical one, it's a biological one. Specifically, we talked with some of the clinicians we worked with, and one of the things that came up as part of the discussion is that Cytoxan is what's known as a prodrug. It's not a cancer drug that's active in and of itself. What happens is it gets processed by your body into the active form. You give it to a patient, their liver processes it, and that's what acts. But what does that mean about what should happen if you give a prodrug to a bunch of cell lines that don't have livers? Nothing should happen. So how did they identify these cell lines in the first place? So we went back and looked more closely, and it turns out they did identify cell lines that were sensitive and resistant, but they're sensitive and resistant to pemetrexed. They got the drug wrong, but who cares because they got great results. So we reported all of this to the Lancet Oncology. The Lancet Oncology wrote back that unfortunately you appear to be arguing about statistical issues to which, as everyone knows, there are no right or wrong answers. So as a result, we do not see the need to publish your objection. We stared at that letter and we moved on. We moved on because actually some other friends of ours who were dealing with brain cancer, glioblastoma, said, hey, they're now looking at temozolomide, which is a drug we use all the time. Can you check their results here? And here's the heat map for gene expression across the cell lines that they're using. And can you match this? And we said, well, we've done that before. Let's try it. And we worked at it and we tried it for a few weeks and we couldn't do it. We couldn't figure out how they were getting these predictions. It was very sad. And we were thinking, okay, maybe we just don't understand this at all. Maybe we missed something. So we said, let's go back to some of the earlier papers and see if we can find what we missed. And in going back to the earlier papers, we looked really closely at the heat map they reported for cisplatin 2 years before. And then we looked at these two heat maps really closely, and we weren't so bothered anymore. And we sent a note to the editors of CCR and JCO saying, these two heat maps purport to be about cisplatin and about temozolomide. One of these is wrong. We'll let you guys figure out which one. And a few weeks later, we got a note from CCR, the editor of CCR, saying, oh, the authors supplied the wrong figure. We'll make a fix. There's no need to publish your objection. And indeed, a few weeks later, the authors supplied a new figure and a new gene list, which did not agree with each other or with the text of the paper. And so what has just happened? Well, in CCR, they have accomplished the neat hat trick of having a paper about temozolomide, drug A, initially with a heat map for cisplatin, drug B, and now with a gene list for some other gene from some other drug, which we still can't identify. All three, one paper, neat trick. Now, all of this, again, is not just the work of a day or a week. No, this is actually now the better part of 2 years back and forth trying to get all this to work. And we're sort of getting tired. So do we care? Why do we keep doing this? Maybe we should just give up. Well, at this point, we got reminded about why we should care, which is a friend of ours got in touch and said, by the way, are you aware that the investigators at Duke are already using this approach to allocate patients to treatment arms in clinical trials? And we said, no, we weren't aware of that. And we looked at clinicaltrials.gov and we read the protocols for the trials. And indeed, we saw that in these trials, in some of the trials, they were allocating patients to treatment arms based upon signatures where, based on the data they published, we thought they had the sensitive and resistant labels reversed. We were not happy. We compiled a list of all the problems we knew about. We wrote it up as a paper, and the first thing we did was we sent it to the editor of a prominent biological journal for informal comment. And his first comment to us was, well, your story's a bit too negative. Can you fix that? And we stared at that and said, you know what? We really don't think we can. So the next thing we did was we sent it off to the Annals of Applied Statistics, which most clinicians read also, so that'll be fine. And the response there was a bit different in that I got email from the editor that evening saying, you know what, I like this paper. I'm going to handle its review myself rather than handing it to an associate editor, which we took as a good sign. And indeed it was a good sign because 2 weeks later, we got a second email from the editor saying, you know what, we've simply decided to accept the paper as is. It's posted online now, which means I'm sending more papers to that journal. I like this. But also it meant that now that our paper was part of the published literature, we could cite it and talk about it, for example, with members of the press. And indeed, just a few weeks after our paper appeared, the Cancer Letter, a newsletter that is read, oddly enough, in the cancer community, ran a front page story saying, biostatisticians at MD Anderson are alleging the potential for patient harm in clinical trials being run at Duke University. Oh, the horror. And unbeknownst to us, folks at the NCI read this story. They read our paper and they looked at grant applications from this same group at Duke and they saw the same types of problems. So the NCI wrote an official note of concern to Duke's Institutional Review Board. This is the group in charge of patient safety, saying, we are worried about treatment of patients at your institution. Can you reassure us? Now, I'm a faculty member at a different institution. There's only so much noise I can make. The NCI is funding them to the tune of tens of millions of dollars per year. They get listened to. So Duke said, you know what? We were going to suspend enrollment in the clinical trials. We are going to convene a panel of experts to re-examine the science and decide whether things should go forward. And we breathed a sigh of relief. We were really upset that it had gotten to trials in the first place, but given that it had, pausing and having experts look at the data, we're okay with that. That's fine, we'll work. And we waited to see what would happen next. And what happened next is that while things were paused, while this investigation was underway, the Duke investigators posted brand new data to their website. What new data was this? It is the validation data that they used to prove that their signatures for cisplatin and pemetrexed work. This validation data is 59 samples, a subset of a much larger published data set, which had 153 samples. But now we know which samples were used, and they say, we use this to prove that this works. So we said, great. We downloaded the entire data set, all 153 samples. We checked the correlations. We checked the numbers that they posted, they said that they were using, and we checked the correlation. And in particular, when we checked and plotted the high correlations, what we should see in this plot, if all of the samples they're using are what they say they are, we should see in this plot 59 red squares, one per sample, all lying on top of this blue line. How many red squares are on top of this blue line? Absolutely none. There are only 43 red squares in this plot. What that says is that for these 43 samples, we can perfectly match the column of numbers that they say that they're using to the raw source data, and the file name and the sample name isn't what they say it is. They got the name wrong. For the other 16 samples, there's no red squares at all. Why not? It's because for those samples, they've already scrambled the gene name so badly, these are no longer valid biological samples of anything. But what we can tell you is that all 59 validation samples are wrong 2 years after they started clinical trials with these drugs. We wrote up a formal report about this, which we then sent to the NCI and to the Dukes at Dean overseeing the investigation. And then we waited to see what would happen next. And what happened next is that in January, Duke announced that it was going to restart the clinical trials. That's because their expert review had finished and their investigation's results had strengthened their confidence in this evolving approach to personalized cancer treatment. We were surprised. And we said, amongst other things, would you show us the data justifying this decision? Duke said no. Duke said in particular, while the reviewers approved of our sharing the report with the NCI, we consider it a confidential document. A future paper will explain the method. we're going to treat the patients this way now, and we're going to explain why it makes sense to do this later. I do not like this. But as it turns out, what Duke said gave us another option. And in particular, we're going to examine exactly what they said in very finicky detail. This is a process known as exegesis, which is a wonderful Scrabble word. But if we look at what they said, let's look very carefully. While the reviewers approved of our sharing the report with the NCI, and if we stop there and we think about what NCI is, the NCI is the National Cancer Institute. It's a federal agency, which means in the United States, it is subject to the provisions of the Freedom of Information Act. We don't have to get the report from Duke. So indeed, a few months later, we got a copy of this report from the NCI pursuant to a FOIA request, and we read the report and we said, this doesn't justify restarting trials. What the hell's going on? And Duke had no comment. And we got depressed because we didn't see what else we could do. So that led a few months later to me sitting in my office in the evening being depressed. And I got a phone call. And the guy on the other end of the line said, hi, I'm a lawyer based in North Carolina. My client wishes you to know some things about the investigators running these trials at Duke University. And I talked with him for a while and I said, you know, that's really interesting material. Here's the number of a reporter that I think you should talk with. And I hung up, and about an hour and a half later, I got a call from Paul Goldberg of the Cancer Letter. And Paul said, I'm going to have fun. Why was Paul going to have fun? Well, about 2 weeks later, the Cancer Letter ran a cover story saying that, oh, by the way, one of the PIs at Duke for these trials used to claim on his CV and on grant applications that he was a Rhodes Scholar, but the Rhodes Foundation disagreed. We know a bit more about this because Paul actually sent a bunch of questions to him about his being a Rhodes Scholar via email, and he CC'd a bunch of the Duke deans. And Anil Poti, that's the investigator in question, said, well, maybe I should talk with him. After all, I was a nominee, and he hit reply to all. So what has just happened? You've had a member of the faculty admit in writing to a member of the press that, oh yes, I've been committing fraud for the past several years, and doing so in writing, CCing the deans for most of the upper administration at his institution at the time he does so. He was placed on administrative leave that day. This story showed up on a Friday. I got a copy of this story. I spammed my friends. I said, I know some of you are getting tired to listening to me harangue about this. I know some of you have talked about writing a letter. Now is politically the best chance you will ever have. So the following Monday, a letter from a whole bunch of rather prominent biostatisticians landed on the desk of Harold Varmus, the brand new head of the NCI, saying, would you please resuspend these trials until the science can be clarified? And Varmus called Duke and said, you're going to resuspend these trials, aren't you? And Duke said, yes. And Varmus called the Institute of Medicine and said, would you consent to reinvestigate this stuff as an independent body? And the IRM said, this has gotten messy enough. Yes, we'll do it. So, okay, they agreed. But before they actually got started, a few other things happened. Specifically, in October, Joseph Nevins, the senior investigator in most of these papers, sent a note to most of his co-authors saying, you know what, we may have to pull one of the papers. Which paper might we have to pull? The one on cisplatin and pemetrexed. Why? Because on closer examination of our database, it appears that 43 of our validation samples were mislabeled, and we cannot confirm the identities of the other 16, which is what we told them the previous November before they enrolled more patients. A few weeks later, Duke officially terminated the clinical trials, announcing that in retrospect, these should never have been run, after which things really hit the fan and a cascade of retraction and resignations, et cetera, began. Now, there were 117 patients enrolled in these clinical trials. Shortly after the termination, the lawsuits began. Around this time, a misconduct investigation was launched, resulting in several retractions and partial retractions from such fly-by-night journals as the New England Journal, JAMA, JCO, etc. This also hit the front page of the New York Times. This also hit 60 Minutes. And because I'm vain and I'm waiting for a brief break to breathe, I'm going to show you a brief clip from that 60 Minutes episode. And this is Joseph Nevins, and I'm going to show you him talking with Scott Pelley of CBS News.
Dr. Nevins discovered that when the underlying data disproved Dr. Poti's theory, the data were changed.
it became clear that there was no explanation other than there was a manipulation of the data, a manipulation of somebody's credentials, and a manipulation of a lot of people's trust.
Manipulated data. These were not errors.
That's correct. It simply couldn't be random. It simply couldn't be inadvertent. It had to been based on a desire to make something work.
Is it a close call, or is it abundantly clear that the data were fabricated?
Abundantly clear.
It is abundantly clear that the data were fabricated 5 years after clinical trials began. Not a good sequence, so folks. Now, brief observation. This story I have just told you is pathological, let there be no mistake. But I'm going to tell you that as a statistician working at MD Anderson, I was able to recognize most of the mistakes in question, off by 1 errors, things like that, because every single one of those mistakes is one I have seen in data sets coming to me at MD Anderson. MD Anderson, a good institution. It's not anything against the institution. It's just evidence of the fact that the most common mistakes are simple ones. They're easy to make, particularly if you're doing data analysis in Excel. Now, if they're in data sets that I get at MD Anderson, I can email the PI and say, this looks weird. Can you send me all the raw data? And I can find it and I can fix it. But this presents a problem because if I don't have that option, in other words, if somebody's mixed up the data and then they've said, oh, well, we've got to publish some of the data because they asked for it, and they publish what's mixed up and I don't have access to the raw source, then we got a problem. And I get worried that the probability of problems in the published universe is larger than we might really want to admit. Now, there is this further question, which is, I've done a deep dive into just how bad things could get. Is this just an isolated case instance, or do we have to have a bigger problem? Well, right at the same time that all this came crashing down, this was when Lee's story came out about, oh, by the way, this is a very widespread problem. And this got the NCI and the NIH really worried because they don't want to support lots of research that others can't use. They don't want to spend millions of dollars on this. In particular, they don't want to have $30 billion a year wasted because we can't reproduce the research. Now, why can't we reproduce it? Well, let's see. Biological reagents and reference materials, this translates to, did you get the labels right? Study design, did you avoid confounding? Data analysis and reporting, did you tell us what you did with code? So that's 90% of $30 billion that can be avoided if we know the problems are there. Now, we were actually asked to say, what do we think people should be reporting if you're going to use this to guide clinical care? And we looked at this and we said, well, actually, if you're going to, at the time that you're going to publish the data or publish your paper, that's the point where we shouldn't have to trust you anymore. So in a clinically guided paper in particular, we want to see the data and we want to see the code because the complexity of the data is now such that written methods don't cut it anymore. But more to the point, there's a simpler way of describing what we want to see when you publish a paper. I want it now. And I'm going to stop that because at the time of publication, that's when the journals have leverage. That's when they can insist upon it. And that's when people will actually do something if the journal asks. I will tell you that if a PI I'm working with is told by Nature that we love your paper, we'll publish it as soon as you post the raw data, that raw data is going on the web real fast. So that's a good thing. Now, after all this, I wanted to review this in the context of one of the reasons why we're here, which is this Rigor and Reproducibility initiative that the NIH started in 2016. If you look at this and you look at some of the grant things that they want in R01s, what do they ask for? Cell line identity verification. This is, did you get the labels right? Some discussion of the experimental design. Did you avoid confounding? Data sharing plan. How are you planning to tell us what we did? The reason I describe it this way is to emphasize that the goal of the NIH is not to make things immensely complicated and onerous. It's to make you think about the basic questions, at least to the extent that you can describe some of the real big problems that could come up in a way that shows that you've thought about them and you can avoid them. And this is actually something I look for when I review R01s, is do they have some discussion of this? It may not be the best. It doesn't have to be the best. But did they think about it and did they avoid the obvious big problems? So I can tell you that we've been doing that for the past several years. We've been reporting data in reports to our collaborators in ways that show code, that get things right and are reproducible because we demanded it, and because we were senior members of bioinformatics, we could impose it top down. So here are some things we did first. And here I'm simply going to tell you that, by the way, these slides will be posted to the GCC website. You can go, you can look at them. And these blue things, these are live links. So if you want to see in detail what things we asked our folks in bioinformatics to do, what code we used, this is a link to the short course that we ran the other year. That's 3 hours of me rambling on in more detail about what we did in R. Here's how we adopted code. Here's what we used. Here's some common structures we used. And even more, here are some details that I would use today. The thing is that over the past 10 years, I've seen data reporting get so much better because the open source tools now make it easier to report data and report code in shareable ways. That makes me an optimist. Now, have we solved the problem? Well, I read through your various questions that you submitted. So let me comment on a few things in 2020. So the answer is no, we still aren't there yet, but there are some things that indicate that we're still making progress, in some ways that indicate that the problem's still evolving. So one of the things is COVID-19. How do we check the data when it's being reported in real time, when we have the potential for a lot more retractions? Well, first off, if you're getting published and it ain't peer reviewed and things like that, we need to be telling the reporters that you need to be a bit more suspicious about these data. And in particular, well, there was a really neat paper by a group called Surgisphere, which talked about the utility of hydroxychloroquine. Some of you may have heard about this paper, which showed up in the Lancet. And you may have heard about the discussion that came up amongst bioinformaticians, where a whole bunch of folks who were chatting about these papers online said, I can't reproduce this. I can't reproduce this. Warner, I can't reproduce this. And if I look at Australia, that looks really weird because they're reporting more deaths from Australia than Australia has had cases. And they went through a whole bunch of this. And the thing is, it's sort of a reinforcing network where one thing about objecting to a paper is if you're doing it on your own, it's sort of scary to do that. But if you've got a group of 30 of you and your friends saying, this is all wrong, and we can point to specific instances why, it makes you more confidence. And that's why the Lancet got a letter from 30 collaborating bioinformaticians from different institutions saying there's no way this is right. And that's why that paper got pulled real fast. So that's another way that reproducibility is emerging. Now, there's another thing that came out this week, and this is a positive story. I don't know how many of you follow the CASP competition. What this is it's a competition to see how well biologists can predict 3D protein structure just based on the amino acid sequence. The fun result of this competition as of Monday is that this problem is now effectively solved. There is an AI that was developed by DeepMind, a former branch of Google, that is achieving prediction results on this problem that are effectively within experimental error. That's really cool. But there's a problem with AI algorithms, which is that even the people who built them don't understand how they work. So how is it that we can trust this? Well, in this case, we can't track every individual step. What are we forced to do? We're forced to say, did this achieve good results on a data set that there's no way they could have gotten good results on if they didn't actually have something real? Believe it or not, this is analogous to something statisticians have been dealing with for decades, which is running clinical trials to see if drugs work. Specifically, they started with the proteins whose structures they were asked to predict. These were 100 proteins whose sequences were isolated in the past year and were in the process of having their structures determined. The structures weren't known at the time the algorithm was asked to predict it, and they still got them right. Pre-specification, blinding, randomization, these tools, if they're followed, can let us extend reproducibility or the idea so that we can even trust black boxes if we think about the data. Now, all of this is just really cool. And there's tons more that I can say about this because I like to ramble, but you've probably figured that out. But I'm simply going to wrap up at this point. I'm going to tell you that I'm an optimist. I'm seeing this getting better, but this is as bad as it can get. So be aware of it. Be aware that simple steps can make a difference. So here are some places you can learn about this. Here are some places you can learn more if you want to dig more into this, because there's even courses about this. There's books, there's greater wonderful things like that. And I do have to do one more thing, which is I have to acknowledge that I did not do all of this myself. The reverse engineering that I told you about, first among equals is Kevin Coombs. Between the two of us, we estimated that we'd spent about 2,000 man hours reconstructing all of this. You don't want to spend that amount of time on analyses that should already have been done. This is not an approach that will scale. We want to do something better. So you can read about that. Furthermore, here's the paper if you want to read it. It's the Annals of Applied Statistics. It's a rather atypical Annals paper because there's only 3 formulas in it and they're all wrong. So go ahead, read it. Even better, cite it, because that's make me famous and my mom happy, and that's a good thing. And I have one last thing to do, which is to thank you for putting up with me for the past 50 minutes. That's what I got, folks. Okay. And I am now happy to take questions or intend to field anything or go from there. Thoughts?
So I think we will. We're running just a little bit over. We'll let, we're going to take questions because we do have time. But Dawn wanted to make an announcement about not logging out of Zoom. Do you want to jump in and say that?
Yeah, I just want everybody, if you could stay in the meeting during this lunch break. I have everyone assigned to breakout rooms, and I'm not sure what'll happen if everybody leaves. So if you could just stay in the meeting, don't leave it. You can stay on mute and keep your video off. I would really appreciate it. Thank you.
And yeah, so we've got about 30 minutes before our next session. So do what you need to do and then come back and we'll start our experimental design panel discussion. But, you know, if Keith is up for it, we certainly can take a few questions if there are some that you'd like to ask. Otherwise, feel free to do what you need to do and then be back here for the experimental design panel discussion at noon. But again, don't log out. Just turn your turn your video and your audio off.
Okay. And as noted, I am happy to answer questions. You can post them in the chat or you can ramble them out loud. And if there's no questions, then I'll refresh my throat and just sign off.
Anybody have any additional questions? Feel free to unmute your mic and go ahead and ask.
I have one briefly. Hi, Dr. Baggerly. My name's Michael Longmire. I'm one of the MD PhD students at UT Health. Mm-hmm. And so I've actually heard this talk before when you talked to our class, but again, it's an outstanding talk and a terrifying but incredible story. So I actually was wondering if you'd comment more about the Surgisphere debacle. And in part because the faculty member had part of his training done in the UT Health system. So I know that was something that was circulating for a while of should we be more concerned with what we've done in-house and what could be the long-term effects of someone who did this did this in the middle of a pandemic.
So I'm actually, sorry, I'm not quite sure what the precise question is that I'm going to respond to here. So.
Yeah, it's kind of just broad. So I guess my question is just, for most of us, what we've seen of Surgisphere is just what has gone out in the news and what may have circulated amongst our own friend groups. I was just wondering if you had any more insights into other aspects of the data besides the obvious ones, like more deaths in Australia than cases reported.
Okay, so one of the things that I will do with many papers is my first sanity check is actually I'll count numbers and say, do they add up to things like from one figure to the next? And I do that sanity check all the time. The other thing that I will look for in the paper is typically some statement about where's the data? Where's the code? Does the paper tell us how we can access it? And does the paper tell us where we can find the code? If it doesn't tell us those things at this point in the game, I'm very suspicious because things this complex require this. There was another thing about the Surgisphere study that looked really weird to me, which is that Surgisphere claimed to have data from essentially all over the world. I mean, hundreds of medical centers. And I've worked with medical record data. I've worked with HIPAA constraints. You know how long it would take to get through the legal rigmarole of getting to use a combined data set from hundreds of institutions across the US and Europe and Australia? And you're going to do this in a few months for COVID and not cite this? No. This doesn't pass the sniff test. So that's something. It's also something of, believe it or not, for big splashy items, it's something where if something comes out in a high profile area and it's big and it's splashy, you'll often see other people trying to test it real fast and do some type of confirmation. And in many cases, the types of successes that they were reporting, if they were using data from all these institutions, some of these individual institutions should probably have had enough data to at least confirm it or check it. And none of the institutions came forward to do that. So that also looked weird. So with respect to the COVID Surgisphere study, that's some of the specific things I can point to. But for studies in general, the issue of, do they tell us where the data is? Do they tell us where the code is? That's something I apply to every paper I read through these days. Does that help?
Yeah, that's the heart of the question I was trying to ask. So thank you so much for going through it.
Any other questions?
I've scared you all. Okay.
I think this means you can have a little break, Keith.
Okay.
Thank you very much.
No problem.
Be
Have a
back
good
soon.
Okay.
Hello, everyone. I want to go ahead and welcome you back. We'll go ahead and get started with the experimental design panel discussion that we're about to start. You've already heard from the other speakers. I will certainly invite them to participate as often as they feel so obliged. But you haven't met our panel yet, so I'd like to invite Robia and Kurt and Natasha all to introduce themselves, and then we will move on with some of the questions and start the discussion. So Robia, you want to go first?
Sure, I'd be happy to. Thank you, Suzanne. My name is Robia Poutler. I'm an associate professor at Baylor College of Medicine in the Department of Molecular Physiology and Biophysics. Should I say anything else? Is that good? Oh, you froze. Please.
Oh, your expertise with animal MRI.
Oh, yeah.
Yeah.
Yes, my expertise is in small animal MRI. We do some work with nanotechnology as well as molecular imaging.
Great, thanks, Robia. Natasha?
I'm Natasha Kiriyenko. I'm an assistant professor at Rice University, Department of Biosciences. My research focuses on antimicrobial resistant bacteria and host microbial interactions.
Thank you very much. Kurt?
Yeah, my name is Kurt Zhang. I'm an associate professor at IBT. So my area mostly is focused on biostatistics and bioinformatics, but I also maintain a small lab, so be able to still do some animal study and mouse study and psychiatry studies, so that I don't forget biological questions.
Great, thanks. So what we've done is we've taken several of the questions that you've submitted in advance, and I'm going to pull those up on a slide. I'm going to share my screen And we're just going to go through those one by one. Again, invite you to unmute your microphone, ask questions, enter into the chat, however you feel most comfortable. Let me see if I can get this going. Okay, can everybody see that all right? All right, so we'll start with this first one, which includes an audience poll. And let's see. For assays that are very sensitive to user technique, how do you ensure that the results are reproducible across different labs? So Dawn, if you want to go ahead, we're going to ask everybody to respond to an audience poll, and then we will show those answers and start the discussion. So you should be seeing a poll. If you can just quickly read through that and choose your favorite answer. Okay, we've got a little over half of you. Okay, we'll leave it for a minute. We're about at a minute. So Dawn, you want to show us the results? Okay, so it looks like the winner is, with 40% of you say that you should repeat at least 3 times in at least 2 different labs. And then about a third of you say if the results cannot be produced by different labs, they should not be published. Several of you thought you could ask someone from a different lab to repeat the experiment and report average results. I do want to mention that these polls were not constructed to have a right answer. They're simply to get the conversation started. So, so we can stop sharing this poll, and then we're going to turn it over to our panelists to comment on this and open it up to you. Dawn, I'm going to let you moderate questions if they're in the chat, because I can no longer see my chat because I'm sharing my screen.
I want to have a quick comment that now we are in a very fortunate time when there are lots of respectable methods journal. We had a couple cases when a technique was particularly tricky, when you need to ensure to mix properly and with certain tweaks or stage animals perfectly to be a certain larval stage for worms we work with. And we found that submitting separate methods paper with a very detailed explanations of how things to be done and what can go wrong was really helpful. This way, in the main paper, we can describe key points, refer to the methods paper, where are lots of side notes and helpful tips on how to perform it properly. And that was really helpful.
I think Natalia brought up some really excellent points. And the other thing I think, Suzanne, if you feel it's appropriate to just share right now at this point, is that a lot of journals also have something known as a key resources table. So that's something that I think does help facilitate reproducibility across the different laboratories. It's also now being required by journals for publication. So essentially what it does is it allows you to identify where you obtained your key resources, such as antibodies, bacterial and virus strains, any identifying features about like lot number, et cetera, maybe manufacturer, biological samples, deposited data, information about cell lines, and it just goes on and on and on. So these are things that I think are really important to also consider for reproducibility across the different laboratories. And I believe that's one document. Suzanne, did we make that available?
Yeah, I'm trying to pull it up right now. Dawn, is there a way that we can, let me see if I can, okay, I think we got it now. Let me just share my screen again.
Okay. Yes. So this one I believe is from Cell. Yep, Cell. And if you just go through, it talks about the different types of things that one can document. It also includes blank areas where you can document your different reagents. And at the end of the document, I believe there are some examples of the types of details one should consider when you document these things. So I think just making this as part of your standard practice moving forward. I do believe there were also some questions about from first year students, how do I make sure that moving forward that my data is reproducible and rigorous? This is a good start. So this is something that should probably be in everybody's, or something similar should be in everybody's laboratory notebook.
This, along with any other resources from today, will be added to the website, and we'll send you that link when we send you the survey. We do want to hear feedback from you all regarding today's workshop. Kurt, did you want to add anything here?
Well, not really. Yeah, I agree with all the comments. So I think the key is, I mean, to be transparent and to share all the conditions and between the labs. So nowadays, yeah, it's so easy. I mean, one person from one lab can do virtual teaching to another lab. I mean, satisfy all the conditions like temperature, like the same cell line, pH, everything. But if the AC is so sensitive that like throughout the complete training, the other lab still cannot reproduce. I really think possibly that methodology is not worthwhile to publish, because to share the society, since yeah, basically it's very difficult to reproduce.
Sometimes it's the factors you don't think about. For example, in C. elegans field, it's common to grow worms in liquid. But if lab is at high altitude in Wyoming, you cannot do it because there is not enough oxygen in the air. So sometimes you need to think what could be confounding variables, temperature, oxygen concentration, that can affect the assay regardless of how careful you are.
So exactly. So those confounding factors, I mean, when we publish the methodology, we should identify if we say, okay, we have some hidden factor we not be able to identify and not be able to tell our co-workers or our labs. So that's raise concern, because that's definitely have a reproducibility issue if we cannot identify those co-variables.
Okay, Dawn, do we have any questions in the chat, or anybody that would like to unmute and add anything or ask a different question?
There are no questions in the chat.
Okay, we will move on to the next one. So this is a discussion question. What are the limitations when choosing a representative figure for a publication? And then kind of following on that, if an assay has a large amount of batch effects, but trends or other statistical significance are consistent across experiments, what principles should be followed other than choosing the best looking figure or the tightest error bars? So.
I can start with talking a little bit of image analysis, because while you can average your quantifications of Western blots, if you take 100 images, you can show quantification, but sometimes people want to show representative image. What we often do in the lab, I'll ask people to quantify about 100 of images to make sure each biological replicate has 30 plus images, and then choose the image that represents the average of the pooled sample best. So this way, when people look at the image and look at the quantification, they have data that very similar to each other, and this way we know we're not lying to even ourselves by showing our best greenest fluorescent bacteria or something of a kind.
So This is a good question. I really don't know how to answer this image question. That's looks so messy. But the batch effect is a big issue in bioinformatics. So in terms of omics data, you have hundreds, thousands of variables, thousands of genes. So between different labs, normally we see huge batch effect even between different person. I mean, from the same lab do the same experiment. So there's a lot of way quantitatively we can normalize those batch effects. So for example, very intuitively we can shift the mean, median, and normalize the standard deviation. And also there's a more complicated way, I mean, machine learning approach to do this. But still, I mean, there's a big chance whenever we try, we cannot normalize the batch effect. So that's just so common. Yeah, we fight with the batch effect every day.
Any thoughts, Robia?
Yeah, I was thinking what Kurt was saying about the batch effects, and I guess one thing to bring up for discussion is one way to try to at least minimize. I mean, you can't completely eliminate, but perhaps to minimize is through the study design, right? So I think you have to consider that even before you collect all of your data, you know, again, you try to design your studies such as you minimize batch effects. So perhaps if you have a group of mice, instead of doing your all of your control mice in your first batch, the best way to minimize this would be to dedicate half the mice to the control group and half to the experimental group, and then repeat that. So that way, if there is something going on with that specific group of mice, or when you conducted that first series of experiments, again, you can minimize the batch effect that you might see as a contributing factor. So I think there are ways, again, to try to minimize this through good study design.
Do we still have Keith on the line? Wondering if he might want to weigh in. Or anybody else in the audience.
I'm still stunned about the representative figure question. I'm not quite sure the best way around it unless you're just repeating until you have something without any smears or air bubbles for a Western blot, for example. But could also just maybe explain that you have some figures that aren't ideal and maybe share those as supplemental. I wouldn't say that's an ideal approach, but at least gets to something that's a little more honest.
Yeah, I think that's actually a really good point. And actually some journals actually require that you show all the gels, for example, that you might be as supplemental data. You might have one representative figure, but then they might want copies of all the data, again, as supplemental.
Is that pretty common, or is that just in a few journals?
I think it's catching on, but we're seeing it more and more. So I think when you're collecting your data, just be prepared to be able to do that if requested by the editor or the journal.
Okay, well, let's move on to the next one. Nothing in the chat, Dawn?
No, nothing in the chat.
Okay. All right, so we have our second audience poll. When is it okay to exclude outliers? When the data does not make sense. If you perform an outlier analysis and the data is shown to be significantly different from others. I'm not really sure what that's supposed to be, Dawn. Can you see what the original slide says?
I can look, hang on.
When the data does not support my hypothesis, when removing the data point gives me a less than 0.05 p-value. Looks like everybody's pretty much agreeing on number 2. We got one taker for number 1. Okay, we can go ahead and end this now. All right, so let's, it looks like everybody's pretty much in agreement. We perform when we perform an outlier analysis and the data point is shown to be significantly different from the others in the data set, right? Any other thoughts on this?
I have a comment. So it's important, if something goes wrong, it's important to write details carefully. For example, you were taking a sample and accidentally added 4 times more dye. So if there is a documented way that the error happened, then it is okay to exclude experiments. Another thing we found really helpful for us, every experiment has built-in blind controls. And for us, in order for experiment to be included, controls have to perform the way that expected. That's a known drug whose response was studied in multiple labs, including our own. And if we don't see an effect, then we'll try to examine what went wrong. And it really helps, especially when junior researchers try to set up complex experiments, because they may forget to add something. So having built-in controls allows you to triage experiments where reagent was not added.
Yeah, I know as a student and as a postdoc, our particular group was not allowed to even do an outlier analysis unless you had details in your lab notebook of the pipette didn't work, or there was too little reagent in this well, or something like you said, too much dye. You had to have a reason to exclude it before you saw the results.
Yeah, exactly. I understand why almost everybody agrees B, because that's possibly all we learned from biostatistical class that do the outlier test and find the outlier. I mean, out of the 3 times standard deviation, then we can throw it out. But that's not the norm practice in every routine lab, because if we cannot figure out how the outlier come from, we should not throw it out. So the reason is just like what Susan and Natalie talk about. I mean, if you actually can determine the outlier animal, for example, it actually has some disease, obviously, and has some overweight, perform very different from others, then you have justified reason to discard. Otherwise, data is data. So you have to take it. I mean, especially most of the experiment we done now, I'm just a small sample size. So even small sample size, then you observe outlier. Part of that's just what come from random. So I didn't say the reason you should do the outlier analysis and throw it out. Unless, I mean, you have really collected 1,000 samples and appeared one about it's really extremely outlier. Okay, in that case, you can reasonably discard it.
Hey, Kurt, in the chat, somebody mentioned the greater than 2 standard deviation from mean, and I know some people are under the impression that's okay. Do you want to comment on that?
2, I would say that's really too small. Yeah. So and normally we teach people like at least 3 standard deviation, and because I mean, think about 2 standard deviation. That's almost covered 95% your confidence interval, right? So that means you have 5% chance, even say the random sample out of a 2 standard deviation. That's too high. So even 3 standard deviation, I mean, that you possibly still have 1% chance, and possibly that's acceptable. But I would still have concerns. So normally, if I look at a standard deviation, I look at a really large standard deviation, like 4 or 5 times.
Anybody else in the audience have anything they'd like to add?
Hey, Suzanne, before we move on, somebody did have something in the chat about the figures, and I just want to make sure that her comment wasn't overlooked.
Okay, can you read that for us?
Yes, it says, one note that was brought up in the morning was that the figure may have taken 5 attempts to make. Should this information be included in the publication?
Interesting.
I was interviewing students, so I missed missed that part of the presentation this morning.
Well, I think the point is if you're having to work that hard to make the figure, It may be a problem.
Yeah, that's for sure. I guess I wasn't quite clear about the 5 attempts to make. It's just like,
Yeah,
is it because
maybe
of
that
the
first thing
data
should
or because it didn't look pretty?
Yeah, exactly. You just 5 times collect the data, or 5 times just try to use the same data, make a figure. It's not clear.
Yeah, I mean, if it's, you're trying to make the figure and it's a function of, or if it's dependent upon the data, then I agree, then that's a problem with the figure.
Okay.
I think we have similar question later on. They come to we make twice attention to reproduce the data. So yeah, when that question come up, we can comment over there.
Yeah, and do also feel free to clarify, you know, just use that chat or unmute your mic. You're welcome to do that as well. Okay, so we had one attendee present this case study. So researcher X publishes a paper in 2012 and researcher Y is subsequently not able to reproduce that data in 2016. The first person moves to a different lab and the samples were discarded by the previous lab. Though the second person followed the same protocol, they were never able to reproduce that data, and they want to know, how should the situation be approached? Isn't the first person responsible for those samples even though they moved to another lab? What kind of recommendations would this panel have for this person?
One
I
of
can,
the options.
oh, go ahead, Natalia. I've already chatted enough. Go ahead, please.
One of the options is, if it's still possible, can researcher X, if the lab is nearby, come and do the experiment with researcher Y samples, or at least watch over video how researcher Y does experiments and point at key steps. Sometimes something as simple as vortexing instead of pipetting can result in a really big difference in RNA stability and then the quality of qPCR. So sometimes it could be something small technical. So I would hope that this all can be parsed to a small technical glitch and start with that. And if researcher Y performs experiments spotlessly, but still outcome is different, then start thinking, could it be different antibody? Could it be media from different companies? So can it be traced to some of the reagents? And if not, then go back into primary data of researcher X and try to figure out what could be the difference.
So, yeah, just short comment. I mean, when you publish a paper, you are responsible to the paper lifetime. So that's period. But even you don't publish a paper and you work in one lab and establish a procedure and move to the other lab, I think you're still responsible to all the procedure you generate and responsible to answer all questions. So that's just a scientific responsibility. So that I believe that's that should be common sense according to any PI's point of view.
Yep. I definitely agree with all of that. And I would just say, in the past, when we've worked with different contrast agents or nanoparticles, when people have moved on and perhaps they weren't, the synthesis wasn't quite the same as before, people have been really agreeable to coming back and walking others through just to make sure that all the proper steps were followed. So I think in general, people seem to be pretty receptive to helping and making sure that people who are in the prior lab are well aware of on how to perform the experiments. And I do agree with Kurt that it is responsibility.
Hey, Dawn, do we have any questions in the chat?
Kelly says, what about differences in data due to older assays or techniques used, but differences observed now or only because of assays that have not been improved over the years?
Actually, I have a comment on that. When my husband was a graduate student, he tried to separate Golgi and ER using established published techniques. And he was following protocols exactly. but couldn't get complete separation. And after about half a year of fine tuning, it turns out that antibody now much more sensitive. So what looked as clear separation before now allows to see traces of cross-contamination. And it's possible mass spec gets better, DNA sequencing gets better, omics technologies improve every time. And sometimes it just changes of what is visible to researcher and what is signal is and what is noise. And of course, that has to be taken into the account.
Okay,
I think we have a couple other comments, Suzanne.
I can't see the chat, so I'm going to have to.
I will definitely read it to you. It's from Sean Chen. How about let another people Z to repeat the protocol before we come to any conclusion? So, I mean, that's not a bad suggestion, and that's definitely workable. And Sachin Thakkar says, how long should a PI keep the sample regardless of the person who did the experiment is present or not? I mean, go ahead, Natalia.
I think there would be a question on how long to keep data later, but in general, according to federal guidelines, everything should be kept at least 3 years since the day of a last payment of a grant. So if it's a funded research, let's say it's an R01 that's 5 years long, an experiment was done in year 1 of R01, then the sample has to be kept for at least 8 years. And different agencies may have different guidelines. For example, if it's something related to HIPAA, then this timeline even increases. So lots of years, if it's a critical sample.
Okay, so we have another poll that will lead us into another discussion. How important is rigor and reproducibility section in an NIH grant proposal? Dawn, you want to go ahead and start that poll? Okay, we'll take about 20 more seconds. Okay, Dawn, you want to go ahead and show us the results? All right, so Roby, I think that this gives us a good springboard. It shows that people are kind of not sure, right? We have almost equal numbers of people that think it's a make or break, required but not heavily weighted as other sections, and that it's a scored section, and some that think maybe it's a tiebreaker if 2 proposals are equal. So I'm going to go to this next resource that Robia has shared and let you talk about that, Robia.
Okay, sure. I'll just preface this with I serve on about 4 to 6 study sections a year, and oftentimes when they give us
sorry about that.
That's okay. Updated training material about the about what to look for in grants. Rigor and reproducibility are actually huge components of what we're expected to score. And the way that you actually couch it or phrase it is very specific. So now when you talk about rigor of prior research and reproducibility, or the rigor and reproducibility of the research, you have to describe it as rigor of prior research. This is something that you find in the significance as well as in the approach section. And it does affect your overall impact score. It is a scoreable component, and it definitely can make or break a grant. We've seen some people actually go into the not discussed pile if they don't adequately address this. But talking about that, there are actually 2 components to rigor when you write your NIH proposals. And for trainees, when you're writing fellowship grants or when you're writing your qualifying exam proposal, you should really keep this in mind. So again, there's rigor of prior research, and what that means is what type of research exists in the literature or from your own laboratory that supports the hypothesis or proposal of what you're proposing to do. But then there's also the rigor of proposed research. And what that means is that you are all your strategies to ensure a robust and unbiased approach. You have to really outline in your approach section that, for example, the studies are going to be blinded, you're going to minimize batch effects, you're going to do so many replicates. And again, this is a scoreable section. But what Suzanne has fortunately done is, this is a slide again from the training videos that are presentations that were given. She will make this accessible to everybody. And somebody, oh, that's different. So again, use these buzzwords when you write your NIH proposals or qualifying exam proposals: rigor of prior research, rigor of proposed research, and then address all of these components. I'm trying to think. So someone does have a question, but it's a little bit different. They're talking about, we had a situation where we requested a plasmid from a lab that had published about it. The lab denied our request, saying that they were still fine-tuning the plasmid. Is that ethical, where there's a publication about the plasmid being fully functional? I think that depends where they get their funding from. If their work was funded by NIH, they are required to share resources with you. And if you go, I think that's not located on this, but another component of a NIH proposal is a resource sharing plan. You have to outline specifically how you propose to share any resources that come from your project, and you are required to share them. So I think there's nice ways to ask a laboratory about that and remind them of that if necessary. And the very, very last option, I think, would be going to the program officer or to the NIH and reporting them as non-compliant. But again, the main goal would be to try to be as collegial as possible and work with people to get what you need and what they're obligated to share.
Yeah, it's also possibly also depends on the journal. So does the journal have specific requirement, like you have to share the plasmid? I believe not, because most journal requires data sharing. Even grants definitely require data sharing, possibly not to the plasmid, but yeah, it's still worthwhile to check. So it's probably not an ethical issue, but maybe the lab just not willing to share. But their explanation is kind of weird when you say it's still fine tuning. So yeah, I think that's a raise some concern.
Yeah, my understanding is if NIH funds them to make a mouse or a plasmid or whatever, they are required to share it.
Mm-hmm.
That's my understanding for sure.
Yeah, that's correct. This is Melissa. It is required on sharing resources if it's NIH funded.
Or NSF doing the same way with regard to data sharing.
Yep. So it's not optional.
And
however,
also,
go ahead.
Some journals also, special methods journals, they make you, the part of accepting publication is that you require to share the tool for the community. For example, some of the nature branch, nature methods used to have that rule that the publishing should be accessible for people, whether if you develop a method or a tool or an instrument, you should be willing to share.
And that goes for software too.
That, the question, I mean, one thing I would say about this, because certainly another element of rigor and reproducibility beyond making sure that you're presenting the best, most reproducible data and that you're publishing that to where people can reproduce it is, what do you believe about what you're reading? And I would be pretty skeptical about somebody that says they have published, a fully functional, characterized, whatever it is, and then they're saying we can't share it with you because we're still working on it. I mean, that to me, and I have some personal experience to back that up, would be a big red flag of, I'm not really sure that I want to put too much of my next 5 years as a graduate student on anything that I found exciting in this publication.
So as a follow-up question or comment, the person who had asked the question said it was actually a lab in Germany and they're not sure what their funding source was. So, but again, I definitely agree with Suzanne about that. If people aren't willing to share their resources, because I mean, it also just benefits them, right? You are able to replicate what they're doing. That's fantastic. They're not willing to share. That does raise a red flag. I agree with what Suzanne said. Chris Ward has a question. He wants to know if there's any resources similar to the ARRIVE guidelines that are more perspective planning based rather than checklist when prepping the paper based. And I think the checklist actually does do that. Those are things you need to think about when you're actually planning your paper. So if you haven't done them, you're probably not going to be submitting in that journal. And actually more journals are more and more requiring a lot of these items in the checklist. So again, just even looking at these checklists prior to or when you're in the process of designing your experiment, I think will definitely be helpful.
Okay, well, let's move to the next one. So we had several questions come in kind of under this umbrella. You know, as more and more of you are starting to do machine learning and AI, what are the appropriate metrics to quantify rigor and reproducibility in AI models? And so we want to open this up to the audience. But Kurt, if you want to take off and start us out on this.
Sure. So I'm really appreciate. I mean, so many people are interested in this question, realize it's a big issue. But this is not a new question. Actually, AI has been used in bioinformatics since the first day bioinformatics established. So traditionally, we have very established guideline and regulation to how to quantify AI models or machine learning model. So for all the traditional approach, supervised or non-supervised machine learning, for example, I think the gold standard is whatever you identify from one data set, you need to use an independent data set to verify and validate your finding. So that's golden standard. But many times we know, I mean, it's possibly impossible to have another complete independent data set. In these cases, we have a procedure like, for example, cross-validation to validate it. So those are well established, but that doesn't mean say have no problem, because it still has a lot of issues. Many people didn't do it correctly. That's a big issue. So for example, when you do a cross validation, suppose you should from the raw data cross-validate until the end finding. But many people start in the middle. So after you normalize the data, after you filter out the data, screen the data, then you start the cross-validation. So that's make your results artificial better, but certainly has a big rigor and reproducibility issue. But that's a procedure issue. Is possibly not people asking here. So in recent years, deep learning is a certain change, complete change in the world. So then the question was very different in deep learning field. So when possible, nowadays we talk about AI, we're more in terms of deep learning. So that question, I mean, is not an easy question to answer. So for many results, I mean, like the example Keith shows earlier, he talked about the AlphaFold. For many of those questions, if the solution you'll be able to validate directly, then it's not a big issue. For example, if you just want to solve a mathematical equation and find the solution, and the solution can put back into your formula and quickly check and find, okay, it works, then it works. So there's no validation issue. So that's something like AlphaFold case mentioned. But for many situations, like if you want to identify biomarkers for cancer, for example, then there's not a, it's basically not easily to check the solution, check the bunch of biomarker if it's not valid or not. In this case, deep learning has a big issue. First, those deep neural network model is really a black box. So we not be able to give any mathematical interpretation, not like a traditional machine learning. So traditional machine learning is well established, well interpretable. But now for the deep learning neural network model, we're not able to interpret anything. So basically, we just throw a bunch of nonlinear function, and we don't know how it works, and then we got results. And in that case, I mean, most biological application will certainly have a big interpretability issue. And certainly there's no way you can cross-validation and check it, since we don't know how it works. And every run, even from same person, every run you basically have different results. And make it worse. So to make those deep learning neural network working, you have to do many pre-processing. So we call it tuning, tuning those hyperparameters. That's probably you have to take a long time before you can even try the model. And that part is really tricky, and basically it's non-reproducible. Non-producible. So everybody knows that. I mean, but that's not a transparency issue, because everybody claim, okay, we have the complete code published GitHub, you'll be able to reproduce it, but how exactly retrain the model, how exactly tune the parameters, that's a tricky part, and that's has a big reproducibility issue. So my expectation is those questions cannot answer, then we have very limited usage for those deep learning model in biological application, unless the solution can be easily checked, like design a drug. So we can easily, after design, which we can check does the drug function or not. But to check the model itself is almost impossible. I hope that answers the question.
Well, that is a super open-ended question. Do we have any other comments from the audience?
So with computational models, what I consider like the gold standard of rigor and reproducibility when I see it in a paper is where the model has been designed on a certain type of data. Mm-hmm. And then after, it's almost like a holdout sample, but it's much more organic, where then it's taken to some very different set of data, like it's in a different type of cell line, or some kind of environment that is like notably different from what it's trained in, and then it continues to produce results. That's when I generally believe what I'm seeing in the paper.
I think that's an excellent point, and it's actually something I was wondering too. Is you build the model, which is great, but then Kurt, do you want to comment on the most appropriate ways to validate the model? I'm not just like
so. Yeah, just like Michael comment. But normally we possibly not use a complete independent cell lines. For example, we have MD Anderson collect breast cancer data for 5,000 patients. So and we test our model and got the biomarker results. And what we need to validate is we need to got independent data, for example, from Baylor. So another 500 patients, and then we rerun the model to those 500 and check does that come with the same conclusion. So that's what I mean independent, because MD Anderson collection possibly have not related with Baylor collection at all.
So here's another follow-up question. When you're validating a model, this might be a way to somewhat address this open-ended question. How do you determine the number of samples that you need to adequately validate the model?
Well, that's not an easy question to answer. And yeah, again, I mean, that's the experimental design, power analysis, but it's not a traditional like a statistical power analysis. So depends on your question. So for example, normally we're interested in on false discovery rate. So based on false discovery rate, we'll be able to do power on that, but that's methodology is most based on simulation. So gave us some preliminary data, we'll be able to simulate that data and generate power and predict how many samples we're going to collect. Yeah, so it's doable, but I would think it's not easy, and not everybody know how to do it.
Yeah, I was just thinking of what you would put into a grant or a study design to convince reviewers, yourself, and the world that you have enough samples or it's adequately powered to validate the model.
Right, exactly. So I think nowadays many proposals didn't include a power analysis for the big data analysis. But normally when I read the proposal, I will show the power curve based on the simulation results from pre-preliminary data. And I never receive any objection about our power. So I think that's a recommended way to do it.
Anybody else have any other comments or questions? Okay, we'll move on to the next one. And so this is kind of a follow on, Robia, of that. I think you had wanted to use that paper for this one. You know, how can we promote unbiased research? And so, you know, if you want to just, you know, chat about that again, but really that I think that that template you provided, and we'll provide it on the website, is a great way. You know, I was thinking about it when we were talking about, you know, when to exclude an outlier. I mean, you don't even think about excluding a data point if it meets what you think it should look like, right? It's only those ones that, you know, fall dramatically out of, you know, whatever curve you're trying to show, or, you know, something that looks, you know, very different. And I think that doing these things in advance, before you making a decision about these things, before you ever even see what the data looks like, is huge. And having that checklist, I think, is very, very helpful. So you make these decisions that if, and, you know, Melissa will talk about this, you know, doing these things prospectively. You know, you want to do this before you ever even get tempted by what the data does or does not look like. And I think a checklist, you know, helps with that. You know, Roby, if you want to add into that.
No, I think that pretty much summarizes it. We went over it. How can we remain unbiased? Again, it all goes to study design. The data is what the data is, and we just have to respect that. You can't massage the data so it aligns with what you hope to see. How can you promote unbiased research? Well, I think just being vocal about that, you know, really when you're at your lab meetings, if people present data, even bringing up that question, it's just like, well, I'm curious, how did you ensure this experiment was unbiased? Was it blinded? How many replicates did you do? How did you determine how many replicates you needed to do? So I just think really having the attitude that you need to really convince yourself and the rest of the world that your data is solid is just something you should be almost have as your mantra. But again, you do have to be unbiased. Follow the template that we are going to provide to you. That's from Cell. That's just one such example, but also keeping in mind just like the NIH guidelines as well.
Anybody have any additional comments?
What I found helpful is go from qualitative to quantitative assessment, not just, oh, this look healthier, this look sicker. But for example, when we're assessing quality of mitochondrial network, this is fully linked. This is partial fragmentation. This is significant fragmentation. That's completely fragmented. And then hundreds of different mitochondria are analyzed and getting binned into the group. And instead of saying these are sicker, we're saying that percentage still looks healthy. Here we have slight damage, and this percentage has severe damage. And then we can do statistics. And we show examples, representative images of each category. And with that, people who read the paper know that how we made our conclusions, and if they want to, they can repeat it easier. So the more you can quantify and show it to others, the easier it would be to reproduce and less biases would be there.
Okay. There's a comment from the chat. I'll read it to you. It says, determining if the data has a normal distribution or should have a normal distribution can help determine if something is an outlier, at least in human data anyways. Kurt, you want to comment?
Oh, excuse me, I'm looking for something else. So, can you repeat the question?
Sure, absolutely. It says determining if the data has a normal distribution or should have a normal distribution can help determine if something is an outlier in human data anyways.
Yes or no? I mean, still, I mean, when you try to determine the normal distribution, that means you have the statistical method keep in mind, for example, t-test. So that's your target. When you design experiment, you already designed your data analysis. Then certainly follow up, you have to check normality. And if not, then you have an alternative design. But I would say, yeah, there's a question associated with your data analysis method, but it's not absolute. So for example, if you propose a different approach and a different test statistics, then normal distribution is not required. I don't know if that answers the question. I can have determine if, well, I think the outlier question, so yeah, in terms of, for example, in terms of t-test, certainly, I mean, normal distribution and based on standard deviation to determine outline, that's possibly logically. But we already comment possibly in biological study, especially your sample size is not big, then it's not suggested. So, and if, for example, if you use non-parametric statistics, then this thing is not appropriate.
Okay, let's see if we can get maybe at least one more in here. So with the COVID pandemic, we've seen the rapid publication of a lot of information heavily covered by media. How can we best communicate this information to the public as data that is still in early stages without losing public confidence in the scientific process? What metrics of rigor still do we need to look for in studies about a disease where very little is known and significant amounts of information is rapidly being discovered? So I'll tell you, we have a team and led by Dr. Ed Septimus that does a digest of all of the COVID literature every Monday, Wednesday, and Friday. Those are archived on our antimicrobial resistance website. We send those out. If any of you are interested in being on that mailing list, you can shoot me an email. It's actually really great. He did one, I think, on November 16th that was kind of a state of the field and where we are. But that sounds overwhelming, probably. I mean, Monday, Wednesday, Friday, but that is how fast this data is coming out. And you've already heard how much of this stuff is bogus. And even doing, a very, thoughtful review and vetting of this literature, we've seen things that, like I mentioned, we archive these on our website. I mean, if you go back to things that were being published earlier, like in even May, June, July, we're already seeing, quite a few contradictions and things that we thought earlier. And so it's a little bit frustrating. And so I think it's super wise. If any of you are interested in this, shoot me an email. But I think it's important to keep in mind that this data is changing a lot. I know I get a lot of emails from the public. I've recently kind of been inundated with several emails about, is it true that I'm good for 7 years if I've already had coronavirus? And I think this is an interesting question for people to ask, because it's, how in the world would you, I wouldn't want to be betting anything on data that we haven't even had this around for 1 year, right? And you want me to make predictions on what's going to happen in 7 years. And certainly the vaccine data is not supporting even that. It's you really have to be very rigorous in what you read and how you interpret it and what you believe. And so any of you, let's ask our panelists what you think about this as well. It's hard to stay on top of all this data.
Yeah. So for me, I think it's very challenging to debunk anything that's coming out in social media or any type of media. I think people tend to believe what they want to believe. I think we just do our due diligence, and if people bring it up to us and ask our opinion, we could say, well, it's very early on, so I think we simply just don't know, but we should just keep this information in mind. What metrics of rigor do we need to look for? So I look for consistency. So it's just like, is it just one report? If it's just one report, I tend not to necessarily believe it. If you see several reports from many different labs across the world reporting something similar, then I start to think this might be true. But I think I definitely agree with what Suzanne said is there's just so very little known about this, and we've only been familiar with it maybe less than a year, about a year now. Like one example would be, I think there were very early reports coming out of Asia saying that your blood type may or may not be protective. And that was very controversial when that study first came out. But now there's several other reports coming out that are kind of saying similar things. So that makes me think maybe, but I'm still not 100% convinced yet. So it's just, again, just keep on your scientist hats and try to review it critically. Look at the study design, look at the numbers, look at the statistics, see if it was blinded or not. And just, you know, approach it as a scientist, but always be skeptical.
I completely agree with Robbie. I just have one reminder. This case study about link between vaccines and autism been debunked so many times at multiple levels. And still, there are a number of people, if you'll start asking on streets, who tell you that vaccination is bad because it causes autism. So I really hope scientists who work on COVID studies are being very rigorous, but don't believe anything until you see that, again, it was shown multiple times, because people may have their best intentions. They may be sincerely making a mistake, but the impact on the public may be dramatic.
Yeah, I agree with both comments. So we do see a loss of rigor in this kind of speedy publications. So for example, for many of the data analysis paper, I can easily tell that methodology has a problem, and those problems easily to pop up in the society discussing. So I mean, everybody see it, and but there's good thing. I mean, when we have this speed of publication and many papers published for the same problem, then we can easily tell, okay, to see the data is, if it's consistent or not. So yeah, I agree with all the comments. Like we should keep an eye, and we should not easily believe one single paper's finding.
I think on the antivirals, and that's my background, I'll be happy when I start seeing one being used by multiple labs as a control. That's when I'll start really believing that, hey, this is the gold standard. This is what we're comparing to. Several people are getting the data. It's just going to be, you know, it's frustrating for us because we're used to being able to pull several years of data and try to make a decision about that. And right now we just don't have that luxury. So with that, I think we're about at the end of our hour. I do want to thank the panelists and thank the audience for all of their input and, you know, for sharing your thoughts with us. And, you know, if you have any additional questions, please feel free to continue to add those into the chat. And definitely invite our panelists to continue watching the chat and responding to those questions if it, you know, it's something appropriate for you to answer. And thank y'all very much. With that, I'm going to introduce Melissa Eitzen from UTMB, who is going to talk to us about data management. So Melissa.
Thanks, Suzanne. Let me just get my slides up here. Can you see those? Suzanne, can we see those slides?
Absolutely, we got them.
Okay, great. All right, well, hello, everybody. Good afternoon. My name is Melissa Eitzen, and I'm here to talk to you for the next, I guess, 45, 50 minutes on data management and resource sharing. My goal today is really to give you some tools in your toolbox to prospectively think about these things and maybe heighten your awareness about a few things, which we've already started today. A little bit about me before we get started. I am the Director of Regulatory Operations down at the University of Texas Medical Branch within our Institutional Office of Regulated Non-Clinical Studies. So what that means in short order is if you think about translational science, where we're talking about basic research bench to bedside, My expertise really lies in those advanced research studies where data is going to be leveraged to design the pivotal studies, and then the subsequent execution of those pivotal studies where data might be submitted by the sponsor to the FDA and scrutinized and looked at for a regulatory decision. So I know sometimes people will say, oh, okay, well, this doesn't involve me. But interestingly enough, especially if you're working with emerging infectious diseases, we actually had an investigator down here a few years ago that was an expert with avian flu and had a lot of preliminary data that was looking really good. And NIH came and visited us and said, hey, we want you to really start thinking about documenting your data more robustly, because we don't ever know when we might be needing to submit that data to FDA to move a particular therapeutic or preventive measure along. And that's what we're really seeing play out now with the COVID research. And then to that end, I got involved a lot with NIH just to talk about rigor and reproducibility than in some of our precursory studies. So that's sort of my background in a nutshell. What I want to speak to you today about are just 4 basic areas. I'm going to give a little bit of introduction from principles and guidelines, spend a lot of time talking about the data lifecycle, do a couple of slides for you on data quality and integrity for some things for you to think about. And then we'll end with a short breakout session with a case study. And when we get to the data quality and integrity, I will ask each of you to have a scrap piece of paper and a pen or pencil ready, because we're going to do a very quick exercise. And for those of you who might have little ones running around, they can actually participate in this too, if you so like. So there really are 4 stakeholders that I like to look at in terms of robust science. And those are the funding agencies, the publishers, you yourselves as researchers, and the institutions. And I put, instead of a biography or references at the back of my slides, the links are actually live on each one of my slides. So this article really talks about or focuses on how the institutions are falling asleep at the job and the researchers are looking to them for help, as depicted in this picture. And while I'm not here to talk about the institutions or critique them, I do think it's important that we recognize that there are these 4 pillars and that it takes all 4 of us to hold up the science for our communities to believe in us, have faith in us, and for our contemporaries to work together as one. So I'm going to talk just a little bit about those 4 pillars briefly. Now, a lot of what we are talking about today started out in 2014 at an NIH public workshop, and this was sponsored by the NIH, Nature Publishing Group, and Science. And the issue, of course, was reproducibility and the rigor of research findings. And the attendees were journal editors, basically from the journals where NIH funded investigators more likely are to publish. The goals were for NIH to identify common opportunities in the scientific publishing arena in order to enhance rigor and further support of research that's reproducible, robust, and also transparent, and then have the outcome be a set of principles to facilitate those goals. So that's what we're talking about today. With the NIH, those principles and guidelines for reporting preclinical research includes those rigorous statistical analyses, transparency in reporting, data material sharing, which is what I'm here to talk to you about. We've already heard some folks talk about imaging. I'll touch on that briefly in my presentation, but I think you're all aware of the importance of that. And your biological materials, such as your cell lines, your critical reagents, antibodies, and the like, your animals or your test system, whether that be a cell line or the animals. Endorsements from the journals. So the NIH is really looking for the journals to be part of that pillar for supporting robust science, to take some ownership in that, and for them to adapt some of the NIH guidelines, which I'm going to talk about as well. So with regards to data and material sharing, the NIH requires that data sets be made available where it's ethically appropriate upon request. And this could actually be during the manuscript review or additionally upon publication. And I think we think about upon publication, we don't necessarily think about during a manuscript review. And this actually happened to me a couple of years ago when I was an author and I was submitting for publication for a grant that that's funded by the Food and Drug Administration. and the editors or the reviewers actually asked for some of the data that I had presented in a table, and I was happy to do that. I was actually really pleased that they asked me for it, and I was pleasantly surprised that they asked, and I was happy to do that. So it is happening, as Robia said. They also recommend data sets be put in public repositories where available. You'll see some of the journals are doing that now. Encourage presentation of all the other data values in a machine readable format. And if you can't do that in the paper, you can potentially do this in some supplementary information or in a repository. Require material sharing after publication, encourage the sharing of software, and require a statement in the manuscript that describes if the software is also available and how it can be obtained. So if you'd like to have some fun in a lab meeting, I highly recommend watching this little snippet. It's a little cartoon, but it has some really interesting points to talk about. It's only about 5 or 6 minutes long, where the bear on the left actually represents a researcher in the lab, and the panda on the right is a physician or clinician that reads a publication of some research data, finds it very interesting, thinks it might be helpful, and calls the researcher and starts asking some questions. And the researcher doesn't exactly always know where the data are. One particular example is a graduate student who's no longer in the lab, who had developed some novel code, didn't document it, and the researcher sort of left to not really know how to answer that. So they're very kind of common issues that I've seen transpire in a lab, but we really ought to think about what should we do or what could we do to prevent that from happening. Going back to those adaptive guidelines that I mentioned, this is where there are guidelines for transparency and openness promotion, or they call them TOP, in journal policies and practices. And I've put a link here in case you're interested in looking at these, because they include everything from transparency, material, research material, citations. And I just wanted to focus on the data transparency, where they have some suggestions about a disclosure about whether or not your data are available and where one might find them, how you might share your data, which we're going to get into in this presentation, and if there are any exceptions or conditions for sharing. We've already heard HIPAA mentioned, so that's absolutely one condition or restraint you might have or requirement for sharing your data. And can the results be replicated using your data pre-publication? And we are starting to see some journals actually require that as well. So we've seen a lot of negative examples, and I wanted to put something positive. So this is actually a really nice example of a publication in Nature on the CRISPR technology. It was published last year. And this is just a section that says data availability and code availability. So it tells you where you actually can find the source data. We're going to talk about source data today too, for the underlying figures, where you can find the supplementary data, and also where you could request the code for that. So somebody asked about resource sharing. NIH considers the sharing of unique research resources developed through NIH-sponsored research an important means to enhance the value and further the advancement of research. And as such, when those resources have been developed with NIH funds and the associated research findings published or provided to NIH, it's important that these results be made readily available for research purposes to qualified individuals within the scientific community. And resource sharing doesn't just mean data. NIH expands this to include samples, critical reagents, model organisms such as transgenic mouse strain, for example, and the data. And my job is to really talk to you about the data. Now, I pulled this definition of data from the University of Pittsburgh library system. And if you ever need a resource on data organization, the libraries are really good at this. They've gotten together and have some really good guidelines for data organization. And I like this definition of data because it's inclusive, not exclusive. It means not only the recorded information, but also the regardless of the form or the media on which it's recorded. So it could be your computer software, records, scientific and technical in nature. It doesn't include information incidental to the award. So we're not talking about finances or an employee's performance file or any administrative or costs or that sort of thing. But they're talking about both tangible and intangible data, the statistics, if you're drawing conclusions, if you have study plans, all of your raw data, your source data, your notebooks, your printouts, photographs, slides, all of these things, IRB consents, IACUC protocols, all of those things really are included in the transparency and the reproducibility of your study. So where do we begin? I like to look at data through a data lifecycle. And if we start at the top, and I'm going to go through each one of these briefly, and we think about a data management plan, the life cycle really starts from cradle to grave. So we prospectively think how we're going to collect our data, where we're going to store or how we're going to store and organize it, how we're going to analyze it, where and how we're going to retain it, what's our retention, how do we protect our data, what are our conditions for sharing and reuse. And while we don't often think about it, disposition is also important. We've talked a little bit in the question and answer session, the panel session, about how long do we retain data. And we heard that it varies on whether or not there are federal regulations, whether there are clinical trials, regulations around that, or HIPAA, whether or not there are requirements from your sponsor. Different sponsors have different requirements, but also state institutions. So the state of Texas actually has retention requirements as well. So most institutions have a records retention department, and you should have a really good relationship with them because they can also be very helpful. And as the case with our public institution, we can't toss anything without reporting that to our actual records retention department to file that with the state of Texas. So it kind of gets more complicated as you think about it, but I didn't want to leave that out. So for the data management plan, I'm going to go back to the University of Pittsburgh again, because I really like this page. Research data management is a term that describes the organization, storage, preservation, and sharing of data collected and used in a research project. And it's on this premise that data are a scholarly product. They're fragile, they're easily lost, as I'm sure all of us can attest to. There are growing research data requirements, and good management helps prevent errors and increases the quality of your analysis. A well-managed and accessible data allows others to validate and replicate your findings. And research data management facilitates the sharing of your research data. And when shared, the data can lead to valuable discoveries by others outside of the original research team. That's really what we are all here for, is to advance science to promote global human health, or for veterinarians to promote animal health, or for the environmental folks, right? You're still looking at the environment, and we're promoting a healthier global environment across the board. So in your data management plan, if you take some time and prospectively think about what your data description and types are there any standards or format for your content? What would your mechanisms for access and sharing be? What are your provisions for data reuse and redistributions? This can all be included in your data management plan, and we're going to just kind of go through those one by one. So when you collect your data, I find it very easy to just do a preliminary inventory of all the data that you're going to be collecting in your study or in your project. I say study, it could be global enough to be a project, which would be a series of studies. Will these be observational? Will they be derived? Will they be computational? What's your format? That's also important in terms of long-term retention, especially for images. What's the quantity? How much data do you have? What are the regulatory requirements or restrictions around that? Are there any proprietary restrictions? And this really comes if you take a contract with an industry. A lot of times they will have some proprietary restrictions around. And I always put owner down because it sounds really funny, but who's the responsible person for your data? In other words, if somebody has a question, who should they go to? And if it's you as the PI, do you have a system that's organized, or do you feel confident enough, as Dr. Ellis said, if he has 30 people that are doing research work for him, you know, and he puts his name on it, he's accountable for that. Well, it's the same way with your data. Do you have a system where you know that you could actually go and retrieve that data at any given time? So I like to think, who's the owner of or the steward of your data on your projects? And source data, which I mentioned earlier, comes in all shapes, sizes, and forms. It can be from the top left if you're doing plaque assays and you take a magic marker and you sit there and you, or a wax pencil, and you write the number of the plaque on the slide on that 6-well or 18-well plate. That's actually the first time you capture data. That's really, truly your source data. So if you don't keep that plastic plate until you actually draw conclusions, then you're transcribing that. And there's always this source of error as you keep further transcribing your data. So the same goes with images, capturing it, whether it's electronic, paper, field notes, or even a printout, in this case of a hematology. And this is where I expect some of you to smile, because everybody that's in the lab has done this. And if you haven't, you're lying. But you've grabbed whatever is close. It's a paper towel, it's a Post-it note, because you're going to forget it. You got to write it down. You don't have the time. I've also written it on the hood, a biologic safety hood, when I'm doing mathematics. I've done that too. And the point is, if that's your source data, then you've just admitted or entered a risk in terms of it might get lost. It might get put in the trash. It might get water spilled on it. And if there's any issues, which there always are on studies, and we go back and we try to resolve them, we really want to try to sort of weed out those transposition errors, as opposed to something that we really need to look into scientifically and say, is this an issue with our hypothesis? Do I need to redesign my study? So transposition errors are very common. This is just an example of a hematology value. I've got a hemoglobin there, a value circled there, 13.6. And if I transcribe that into, say, an Excel program or some kind, or Prism, and I have a key that sticks on my computer, for example, and the 1 doesn't transpose over, and now it's a 3.6. I've now changed a normal hemoglobin value to an abnormal hemoglobin value. That's going to impact my study outcome. And I may or may not catch the fact that this actually was a transcription error. So thinking about organizing your data, think about where you're going to keep it, whether it's a physical location for paper data or an electronic data. Think about your transcriptions of your source data, the accessibility, security. And I put change control on here because the image on the bottom right is the autosave function, again on Excel. If you're using Microsoft 365, which is wonderful for collaboration, because you can share files with collaborators that are offsite or even within your own team as we go through the pandemic, if you have that autosave function on, then it's automatically going to rewrite to your same file. And if you have any kind of transcription errors, or as Dr. Baggerly showed, just a copy and paste error, you're rewriting over an original file and you have no way to go back and look at that. So these are things that I just put as aha moments. Think about if you have an Excel spreadsheet that you're going to continually add data to during the course of your study. Do you save these on a daily basis? Do you have some sort of a stopgap where you could actually go back and recreate the study if something happened? And that could be as simple as an autosave and rewriting over data that you didn't intend to do or didn't realize you're doing. Managing electronic data, if you have the ability to turn on an audit trail, that's great, because that actually will track a lot of what's going on behind the scenes, particularly if it's associated with an individual. And attributable is something that's important, because we often in the conclusions of studies have questions, and we really need to, we want to go to the person that has the most information or the most knowledge about what happened at any given point in time. So being attributable, knowing who was responsible for collecting or transcribing that data, is extremely valuable. Also protecting our data from security and encryption. And the software compatibility is interesting, because I share the story, that true story. I was in a board meeting one time several years ago, and somebody was using a laptop that they had borrowed, and we had somebody remotely that was participating via Skype. They had to download this Skype wasn't an application at that moment, at that time. And what it did was it disabled the copy and paste function in Excel, unbeknownst to that person. So sometimes there are random things that can happen to another software that's on your computer. So be really careful. I know a lot of folks who like to do data crunching on their Macs. And so they'll have a personal computer that they do that on. And you really need to think about the traceability and who's doing what and what might impact your data that you're not thinking about it. So just think about backups, automatic program updates, automatic pushes. If you're migrating any data when you have updates, updates to Microsoft, I've seen it change significant digits on Excel and on other statistical programs. So just be cognizant of that. And if you work with large data sets, you may be already familiar with a checksum application. There's some free applications on the web that you can use, but it basically does an algorithm on a particular file and it comes up with a big long number. And if you run that again, you'll come up with the same number if you use that same particular function. And what's nice about that is if the number is different, it means that there was a change to that study file, even if it's just a space. So I see a lot of folks that have a lot of big data, like the CRISPR technology, where they'll actually save their data in a file, run a checksum, save that checksum in the file. And that's how they assure that that's the original data set. And then they'll make a copy of that. And then they'll do the manipulations off the copy of that. And that's how they'll assure the integrity of their study or of their data. So here's just an example of getting organized. The one on the left, I pulled actually from the web, which is really nice. You can use a series of numbers and letters to represent the file types. And the one on the right is an example of study organization that we've used in the past, where you just create this for every project, where your files are organized the same. So if you want to go in and you want to look at data from a previous study, you're going to know where to look because you organize them in the same way. So you might keep your original study plans separate from your IACUC or IRB approvals, any kind of project management, Gantt charts and the like, just some things like that. And communication is also really important to keep, especially if you make any kind of decisions about changes to your study. Maybe your NIH project officer asks you to change something, which I've had happen to me a lot in the COVID pandemic. And you want really to document why these changes occurred and who authorized or who directed you to make those changes. Because I guarantee you won't remember 2 or 3 months down the road when you're trying to summarize your data and publish. Analyzing, again, prospective thinking, think about methods to reduce those transcription errors, define your inclusion and exclusion criteria up front, and that reduces your bias. So if you talk about experimental design and maybe you're doing some PCR analysis and you're going to do those samples in triplicate, define triplicate, define what your outliers are. So when would you reject one of those 3? What are your conditions for repeating, so that you don't use that scientific intuition that says, this doesn't look right, I'm just going to rerun it until I get the result that I think I should get? That's bias. So this is what prospective thinking really means. We've talked about statistical plans and retaining any methods that you use to allow for study reconstruction. We talked about images. I pulled this from the Office of Research Integrity as an example. And I don't want to take a lot of time to go over this, but the top left one is actually the control, and the protein A is the same image. It's just got a different resolution on it. And so it was actually presented in a paper as a control versus protein. Figure 2, if you look at M1 and M4, those are flipped along the x-axis. They're actually the same image, only inverted. So I actually have the answers to those if you want to see some there. But the real goal there is, if you're doing image, and we all manipulate our images because we're trying to get the best image that we can, please document the changes and retain that unprocessed image so that you can actually go back to that raw source data and say, well, this is the source image. And this is just sort of a color cartoon from the Office of Research Integrity that gives you an example of what happens if you over analyze in terms of contrast and saturation, and then your background cells in this particular example disappeared. So retention we talked about, or you guys listened to during the panel session, so I don't want to take a lot of your time there, but really you need to think about where you're going to store your data and how easily it could be retrieved. And then for data sharing, how that might be done, whether or not it's can be done in an email with a link, whether there's an online repository, a supplement to your publication. Think about sharing agreements, so conditions for sharing, especially if there are some information in your data that could be disclosed in terms of HIPAA compliance. It's always a big one. And maybe it would be mixed. Maybe you could provide a link to some data and then go to a repository for something else. Conditions, exclusions, and instructions are really important. I was looking at some data for a large set of data for an investigator. It was very intuitive to him to know what it was. But when I went into his data files, they were very well organized, but I really didn't understand how he had them organized. And it took me 15 or 20 minutes to figure it out, and I did, but he could have saved me a lot of sort of pain and anguish if he would have just sent me an email with a few simple instructions that said, you know, study A is found in this file, and this is how I go about it. Here's just an example of a data sharing plan. So there's actually a template on the NIH website if you'd like for that. And reusing is essentially data sharing, but that's really if you're going to reuse it, thinking about any conditions and instructions if it differentiates from the original. So in the last section of I've got 2 or 3 minutes and then I want to do the breakout. I do want to introduce the ALCOA principles. For those of you who haven't heard of them, it's an acronym which stands for attributable, legible, contemporaneous, original, and accurate. And it means I really need to know who collected that data, and I want to do it as quickly as possible. Because if I ask you, what did you have for dinner last Thursday night? Most of you can't remember. So it's the same way with writing down what happened in your laboratory. And then consistently, your records should be complete, consistent, enduring, and readily available. So I'm going to take 30 seconds now to do a really quick exercise. So grab a scrap piece of paper. If your kiddos are by, they can do it too. I'm going to give you 30 seconds. So it's going to be really quick. And I want you to describe the image that I'm going to put on the screen. So are you ready? Go. Just describe it. Fifteen seconds. Five seconds. Okay, stop. So if we were in a face-to-face course, we would talk about how we describe this cute little mouse eating a carrot or a cracker or a Cheez-It. It's always fun to see what people think that is. And there are some nuances and some differences, which is important because we're subjective when we're writing our observations, and we should remember that. But let me ask you, did any of you date and initial your observations? And if you didn't, can you quickly do that now? Can you just put your initials and date as if this were an entry to your data? And when you did that, look and see what format you put the date. Did you put it as month, day, year, or as date, month, year? And I find this to be really important because I see that if you're folks that are from Europe will write it one way, folks that are from the Americas will write it another way. And if you're using numbers, this can be a real problem when you're trying to reconstruct a study. So I always advise people just put like AUG for August instead of 8 for August. Here's another important resource for you guys, Quality Practices in Biomedical, Basic Biomedical Research. It talks about without documentation, the process is actually meaningless. And there's a lot of really good, helpful information there for research labs. And to go back to the definition of data, remember that our scientific data includes both intangible and tangible data. So to conclude my presentation, I always go back to Dr. Ellis's and the Begley-Ellis article. And in that, where he talks about the 53 landmark studies where they tried to reproduce them and only 6 could be reproduced. That's only 11%, by the way. These are the 4 things that those 6 confirmed studies had in common. They addressed and included controls. They confirmed and validated their reagents. They prospectively in their design addressed investigator bias, and they described their complete data set. So with communication, I like to show this really cute real life example. I saw this email come across my desk. It says, I'm going to miss the March 3rd Friday call-in. And if I look at March 3rd and Friday, March 3rd in 2019, that was not a Friday, it was a Sunday. And what the intention of this was, really, they were going to miss the third, it was a reoccurring meeting that occurred on the 3rd Friday of every month. And they meant to say, I'm going to miss that March 3rd Friday call. But in this case, it looks like it was March 3rd. So be careful and be mindful that sometimes what's clear and transparent in your own head is not necessarily what someone else might interpret it. So it's a fun exercise to do in a lab meeting, is to exchange lab notebooks and see if you can actually understand and conclude what your colleagues are doing. So in closing thoughts, get organized, remember data stewardship throughout your data lifecycle, prospectively put together a data management plan, prospectively love your data. Don't forget to spend some time on your data. Actually, I had an NIH inspector tell me that you're not loving your data as much as your scientific plan. You really need to think about and protect them, and implement the Alcoa principles. And with that, I'm going to stop sharing, I guess, Suzanne, and do we have time to go into the a quick, maybe 5-minute
Yeah. Okay.
Or do you want to just, do you want me just to do this all together?
Let me see. We're trying to figure out how are we ahead of schedule? We've got until 1:50.
Yes.
We're good. Let's do a 10-minute breakout and then 10-minute reporting. So Dawn, do you want to go ahead? Now wait. Now Melissa will. We need to have the case study up, or we can put it in the chat.
I can put it up.
Okay, and so we will also, Dawn, we probably need to add that to each chat room, or can you send a message?
I will try. That is a good question. I'll figure something out. I'll get it in there for him.
Okay, all right, so you'll have about 10 minutes. Remember, you're going to identify your recorder and your reporter, and you want to do that quickly because 10 minutes will go by very, very quick. And then Melissa will call on you by group numbers. Okay, so
yeah, so take note of the room number that you enter, and I'll pull like randomly about 3 groups to share, and there's no wrong answers, so just talk amongst yourselves, and I think they'll time out, right?
Yeah, they'll time out, and also remember that your recorder is going to take down all of the notes, and then those are going to be emailed to me or Dawn. Okay?
Okay, here we go.
Good luck. Great, Suzanne, do we have them all back, John?
Looks like we've got everybody back. So I know that some of you did not get this until it was almost over. I'm going to call on the ones that I know had it a little bit longer than some of the others. So let's start with group 1. Why don't you report out on what you found? So whoever the reporter is for group 1, go ahead and unmute your mic.
We didn't really, I apologize. We weren't really clear on exactly what we were supposed to be doing with this case study. And I wasn't nominated to be the reporter or anything, but basically we talked about, if we're sharing data from a human studies, from a study that involves humans, then you have consent issues where if you have broad consent that's been taken from the subjects prior to the original study, then you're able to share that data for a secondary study. So there's obviously HIPAA regulations that would be involved in this. So to share that data, you'd have to, there'd be regulatory considerations. So that's about as far as we got.
That's good. For only having it for probably 2 or 3 minutes with very little instruction. That's great. My apologies for technical glitches.
So we're not sure, like, you know, maybe there was a different way that you wanted us to look at this as far as like how we actually share the data, as far as like what kind of medium or format. We weren't
Right.
sure exactly which direction to go with this one.
Right. There was actually a really long case study. We truncated it so that we could share it with folks. So we'll make a note to improve that for the next class. My apologies.
How about group 5?
Sure. So we talked kind of similarly about how not really like how like you would use media to kind of transmit it, but sharing in terms of privacy concerns. So we were talking about how like the screening could be like it's sensitive and like confidential information. So there's a level of de-identification that needs to happen if you're going to share it. So all of the like identifiers that can be used to trace this data back to a particular person, like name, address, like the dates of certain procedures and stuff like that, need to be removed from the data if you are going to share it. And then sharing it with people who have had the like proper training and have gone through like the human subjects training and all of that. And then also only with the people who need to have that information. So not like sharing it widely, but like designating the people that need to have access to the data and kind of focusing how widely that data gets spread.
Yeah, outstanding. That's really good. I like the fact that you put in about training, right? Conditions in terms of will the individuals you're sharing the data with have the appropriate training to understand what they can and cannot do with the data. So that's a really good point. So Suzanne, I think we probably should stop there just because of time, but I want to go ahead and show the, I don't know if I could, does it advance? It's not advancing now. It's crazy. Okay, well, I was trying to advance my slides, but I don't know why it's not doing that. Oh, there we go. Okay. It's just a little slow. So I actually pulled this from the NIH website strictly because I wanted to share an example that they put forth. So you can see in the grayed out area there that there was actually a little bit more information to the study. We just truncated it because we weren't sure how to share it with you all. So we'll make a note to improve that for the next course. So my apologies for that. And while there are no right or wrong answers, this was an example of how you might put some conditions. So we will make the data and associated documentation available to users only under a data sharing agreement that provides for a commitment to using the data only for research purposes and not to identify any individual participant, a commitment to securing the data using appropriate computer technology, and a commitment to destroying or returning the data after the analyses are completed. So I thought this was a really good one because it just gives you an idea of the forethought that you might really need to put into it if you're talking about sharing your data. So to take that full circle, resource sharing is a requirement from NIH funding and from a lot of other funding agencies as well. But for this particular one, it's for NIH. And because of that, you really need to think, well, if I'm going to share my data and if there's any kind of HIPAA regulations or personal information that might be shared, how might you comply with those while also complying with the NIH requirement to share your data if it was requested either by a publisher or by another scientist. So those are the sort of the things that I wanted to bring to your attention. There's my contact information there if anybody has any questions. And then I put an extra slide in there that I like to use for how to start and keep a laboratory notebook, which I think is really important, has some really good useful tips. And so I'm sorry I don't have time to answer any other questions. We're sort of late for the next speaker, but thank you for your patience on the technology, and we'll improve it next time. And I hope that you learned something, and I wish you all good luck in your labs and your careers and your research, and please stay safe.
Thank you, Melissa. And these slides will be on the website, so you'll have access to those. But with that, we will turn it over to Stacy Gorniak, who's going to talk about publication and reporting.
All right, so today we'll be talking about publication and reporting, talking about some of the issues that we have as investigators dealing with lags and things such as publication and reporting. And to start, I want you to take 1 minute, a pretty reflective moment, right? And so think about examples in which you personally have exhibited some avoidance behaviors, such as procrastination with respect to publication and reporting in any of your current or your past research projects. So you probably didn't think that you were signing up for a reflective moment this afternoon, but just think about this and what are the feelings that come up and what are the thoughts that you have around this, okay? What we're going to be talking about today is getting down into why you have these behaviors and what are the types of things that we could do to counter these behaviors, but also some unintended effects of these behaviors also. So avoidance motivated behavior is behavior that we basically exhibit to distract ourselves from doing some type of task that's associated with an unpleasant emotion. Typically that unpleasant emotion is fear. In the psychological behavioral world, this is also known as approach avoidance. There are 3 main types of behaviors that you might exhibit. One is complete avoidance, where you no-show at a presentation, you don't submit documents, or you just simply quit a responsibility. Another form of this avoidance could be escape avoidance, in which you leave early from a commitment, you end a talk abruptly, or you hide in your office. I do actually know people that have decided to hide in their office because they didn't want to do something. And then lastly, there is partial avoidance, right? Where you do something such as daydreaming, or you avoid eye contact, or you web surf, right? Instead of doing the task at hand. And basically, when we have these types of behaviors, they're associated with things such as worry, anxiety, and panic. And when we do these avoidance behaviors, they don't reduce our anxiety. If anything, they actually fuel it or cause these difficult situations to snowball. And so there's a lot of cartoons and funny drawings out there that talk about avoidance motivated coping. And the number one way that we do this really is in procrastination. PhD Comics has many, many different cartoons on this phenomenon. There's a multi-panel, I think it's a 12-panel cartoon right there that's a field guide to procrastination. I already know who I am. I am definitely the cleaner and the snacker. So it's important to know which ones you are. Modern procrastination. So this is a cartoon I found that shows the 4 horsemen of modern procrastination, right? So naps, snacks, social media / Netflix, and minor chores, okay? These are all things that we struggle with, especially with COVID, being home a lot and looking for distraction behaviors. And so when we have this avoidance motivated coping, it's doing something else in order to bring a momentary relief. When we talk about avoidance motivated coping, we're not talking about straight up maladaptive behaviors such as data fabrication, in which data were never collected, they're just made up, or data falsification, right? Where data are legitimate data may be altered. Okay, these are not necessarily what we're talking about. These are very extreme behaviors. And if you need more information about what those specific definitions are, what examples of those are, I know earlier today, some of my colleagues definitely talked about them, but the Office of Research Integrity website has a lot of information on this. They have recent cases that you can look through regarding data fabrication, falsification, and other forms of research integrity violations. So the goal is to not let your avoidance motivated behavior get into something like fabrication or falsification, right? And so why do we exhibit these behaviors, these avoidance motivated behaviors? Usually we have some type of fear of failure, all right? There could be a lot of pressure to produce, right? Publish or perish, right? You might have pressure to have high impact publications, whether it be in your laboratory, your institution, or just generally in your career. There could be issues with employment or visa pressure. That's definitely something that I've heard of in terms of some of our international students. And so some of these things can just really get to you, right? And really just cause some bad situations. Other things that could be driving some of this is just difficulty with time management, okay? This is something that is not uncommon, especially as you move into more advanced levels of your career. So when you're a student, you're a postdoc, you're in the lab most of the time, you're not dealing with a lot of the administration, but as you move towards more senior roles, you just get more and more administration, basically, right? And it really just takes a significant amount of my time and effort into administrative roles that you may not have accounted for. Also, some of the deadlines that you may face can have conflicts with other commitments, whether they are job-related commitments or personal-related commitments, okay? Beyond that, sometimes we have these avoidance-motivated behaviors because we feel like we have a lack of progress, right? So often, and this is, academic guilt, we feel like we are not productive enough. And especially with COVID, a lot of people are talking about this, they're feeling that they're not being productive enough, they're not doing enough. And that can be very, very, very paralyzing. So if you are in that mode right now, you need to recognize that and be a little bit more kind to yourself. Sometimes our lack of progress is because we don't have the information we need to move forward. And sometimes our progress is actually just a moving target, right? So, things just change in research, right? We have to pivot, we have to do something else. So these are all just different things that can contribute to these avoidance motivated behaviors. Okay. And avoidance definitely impacts publication research in many ways, such that you can miss your deadlines, you can have delayed publications, late reports to any type of regulatory or funding entities, whether they be internal to your institution or external. And they can definitely impact promotion and tenure as well as career advancement. So these are some very, very serious considerations, just in terms of personal stuff, right? But also we're going to talk a little bit later about what the impact is to the evidence base in terms of avoidance behaviors. But before we do that, I want to talk about, what are considered products of research. So typically when we're talking about products of research, the number one thing that comes to mind is typically our peer review publications, right? There are other things that are considered products of research, such as conference papers, podium presentations, abstracts, conference posters. If you have any type of technology development, intellectual property, that's also considered a product of research, licensing agreements, databases, physical collections, software, instrumentations, prototypes, inventions, and whatnot. Those are all considered products of research. So it's not just, one line on your CV that is a publication, right? There could be an entire body of products of research. So who typically does the work? Typically your PI co-Is, co-PIs are going to be your senior authors, but really it's your postdocs, your research assistants, your grad students, maybe even undergraduates, depending on the institution, and your lab techs that are really doing a lot of the work, right? So the PI is going to be spending a lot of their time managing the group as well as dealing with administrative issues, right? And so that's why a lot of the work in the lab is done by the junior scientists. And so the one thing I do want to acknowledge right here, because you're probably sitting there going, oh my goodness, I'm the only one who's procrastinating. The reality is no, okay? Avoidance behaviors exist at all levels of science, right? It's ubiquitous. I experience it. I mean, 2020 has been pretty rough. So those of you who don't know much about research reporting, here's just a little bit of information. There's a lot of different administrative levels that you might have to report your research to, right? Whether it be your department, center, or your university, they're going to want to know what you're doing, right? So your annual reports. You're also going to have to provide continuing review information to your IRB or IACUC boards. Any federal regulatory bodies such as the USDA or Food and Drug Administration or the FDA, but also when you receive grants funding, right? Whether it be internal or external, they're going to want to know what you do with that money, right? So you're going to probably have annual reports to those funding agencies. And so here's just a cute little pie chart. This was found by one of my friends several years ago about the difference between your basically a postdoctoral situation and when you become a PI, just the time that you used to spend in the lab and doing research, that time just gets divvied in so many other ways that you don't even think about it when you're a postdoc and a graduate student. All right, so when you have to do these annual reports to your funding entities, NIH has a standard format, and that standard format has been adopted by many other private entities. That they might have some slight modifications to it, and these are typically annual reports. Usually only the primary investigator or some type of delegate can initiate and submit these. So the PI does have a lot of responsibility to make sure that this gets done, and it gets done accurately and in a timely basis, right? Some institutions have really, really great resources around getting these done. They might have entire departments or staff members, administrative staff members to help you with this. Other institutions may not. I know that at the University of Houston, this has been an issue, and over the past 10 years since I've been in Houston, this has definitely changed. They are getting more and more people onboarded to help with these administrative issues. So, but it's really important to know what your resources are. Are you the one that's going to have to be doing all this, or is there somebody that can help you at your institution? That's really important to know. So when you're filling out these annual report forms, you're going to have to tell the funding agency what were your major goals. Were they accomplished? Were there any training or professional development activities that were provided from this project, either for you or for your trainees? Have you been able to disseminate your information to the community? And what are you going to do next, right? What are your next goals and accomplishments that you want to achieve? So that's the fun part, right? So that's where your publications and products of research come. You might also have to produce patient or animal safety reports, depending on what type of project you're dealing with. You might also have to report on collaborative efforts, especially if it's a multi-site project. And some institutes want to know what impact you're actually having on the field. In terms of roadblocks, right? In your annual reports, you should have an opportunity to present any project challenges or changes that you've had to implement, and actually provide a rationale as to why you made those changes, delays, and what action plans you have to resolve them. For those of you, like for example, my lab right now is completely shut down due to COVID. I can't see a patient, I can't touch them. They're not even, we don't even bring them on campus. When I file for my NCE, I'm going to have to tell them, hey, my university was shut down in terms of human subjects research. I can't even see a patient. So this is the reason why I don't have any data. The last data I collected was March 3rd. Okay. You're also going to have to produce financial and budgetary reports, as well as modifications and future plans, particularly if there have been changes or challenges. You need to provide a plan as to how to overcome this. So one of the things that we also need to talk about here is that when we're doing our publication and reporting, there are some core standards that we need to consider in the products that we produce, whether they be annual reports to funding agencies or just scientific proposals or manuscripts out there within the evidence base. And one of the most important things that we need to talk about is randomization, right? Have you actually randomized your sample, and have you shared that information as to how they were randomized or stratified randomized or whatever you did? You also need to talk about whether or not you had blinding if you are doing human subjects research in particular, or even animals research. Did you do a single blind, a double blind, a triple blind? There's information about how to implement each one of these on the web. I would definitely recommend looking at these if you've never heard of them, right? So your randomization, did you report that? Did you report your blinding? Have you reported your sample sizes in terms of estimations, and how did you get there? You really need to provide evidence that your sample size was appropriate for your measures as well as your aims, right? One of the most common things that I see in grants is that studies that are underpowered, they might be powered for one aim, but they're not powered for all of their aims, right? So you need to be mindful that you are powering to your weakest phenomenon, right? And just being very open about how you came to those numbers. There's a lot of different ways that you can report this, whether they be in manuscripts or within grant proposals themselves. You also need to report your data handling, right? And so this was what Melissa was just talking about. You know, how do you secure your data? Were there any specific primary endpoints or secondary endpoints? Did you have any missing data? Was there attrition? how many replicates did you have, right? So these are all things that you need to make sure that you are being very straightforward about within the products that you are producing. So like I said, sometimes we have these things that happen in terms of the laboratory or even larger in life that can really cause us problems in publication reporting. You know, maybe you're having difficulty running a project or something came up and that just, you know, whatever you were planning on doing changes, right? Maybe your staffing situation is different. You still have to produce what you promised to produce, right? To the standards that you said you were going to produce them. So, you know, you need to have a plan as to how to deal with this. And sometimes when these things happen, these delays happen, or there are other challenges, it can be really, really difficult to organize all this information. Communication challenges, time conflicts and whatnot, or even problems with institutional support. So just keep all of these things in mind when you're doing your projects and how you're going to deal with them in order to just maintain, you know, high quality products of research. So if it's also hard, why are we even going to bother? Well, I talked about the negative consequences to our personal selves, right? So it could be withdrawal of funding. It could be an inability to support current and future projects as well as your own staff, but also in terms of your own career advancement, right? So there are some very personal consequences that can come out of this. But in addition to that, there are negative things that actually can happen within the evidence base, right? So the number one problem that we see are different forms of biases, right? Get into our evidence base. So reporting biases are a major one that we see, right? And so this is where we have selective revealing of information where you might report one thing that you found, but possibly not another, or spinning of undesired results. Okay, so this is something that definitely happens in publication or reporting, and we need to be mindful of this and that we're not doing this. Publication bias is another form of bias that we might encounter, right? So this is essentially non-publication of results, and typically positive or significant results are preferred, right? And so when we have non-significant or negative results, they don't get published. We also have issues with time lag biases where we are very quick to publish very exciting results that might not be the full results, but those negative or less exciting significant results, those might take quite a bit longer actually to get into the evidence base, right? Citation biases, we tend to more frequently cite positive findings, and that could be associated with the fact that they are just generally out there more than negative and non-significant findings are. And so there's definitely an overrepresentation of positive findings. We also have language biases. Typically, positive findings show up more in English language journals, whereas non-significant results may be published in non-English language journals. And so it could be very difficult to find some of these negative or non-significant study results. And it definitely impacts these meta reviews and systematic reviews that are produced within each field. Other types of biases, outcome reporting biases. So expectation biases can be reinforced such that selective reporting of results are basically just reinforced. There are situations in which hypotheses might be modified to actually fit the results. And some type of negative result might not be published, right? So this is just an additional layer of data suppression, which can be problematic in almost every single field. Other forms of biases, location biases, where we tend to, the studies with really exciting, positive, significant results tend to be in higher impact factor journals, whereas the less exciting ones, those less exciting results are just going to be in journals that don't have the larger impact factors. Okay, so this goes hand in hand with people really pushing to publish in really high impact journals, Cell, Nature, Science, things like that. So we just need to be mindful of that bias. We also have funding bias, right? And this could be the tendency of a scientific study to support the interests of the financial sponsor. So for those of you who are already PIs, you have to sign off on conflict of interest forms to make sure that things like this don't happen, because we have had issues with this in the past. We don't want to introduce an expectation bias into the evidence base, all right? So things can get very, very sticky when funders want a certain outcome. And so this is where your conflict of interest office really needs to be involved. And so how can we address bias in publication and reporting? This is where you need to be able to build a case where both positive and negative findings can actually provide information to the evidence base, right? Don't try to suppress your results. Don't rush or delay publication, but also don't dual submit. We can really find those these days. We have much more powerful search engines in order to find those. We also want to consider the impact factor, the scope, and the audience in which we are submitting our journal articles to, right? So are we submitting this to the right place? Are we submitting it to the people that really need to see it? And if you have data sets that you want to do different things to, you need to make sure that you're not actually just doing dual submission, but that if you have a completely new form of analysis, that gets reflected in like some type of secondary paper from a project, right? And so we don't want to do the dual submit situation. Try to balance the story you tell with respect to citations. And NIH actually has a really, really great website that talks about the different rigor and reproducibility guidelines that they expect scientists to follow. And the link is actually here within this PowerPoint. Other things, don't withhold critical information. And one of the things that we do see pop up around this is the discussion of authorship and team expectations early on in the process. I actually heard about an issue around this recently, not in my group, but you know, managing expectations as to who does what on the team and who gets credit for what is something that you as a PI really need to be mindful of, because when you don't have these things set early, it will cause problems later on, right? It can definitely cause issues with time lags, production issues, and just poor team dynamics. So please make sure to just be very clear with your team as to what their roles are going to be and what outcomes they should expect. Be organized, but also set realistic goals, right, for yourself and your team. Especially with COVID, things are just at a very different pace. They could be, in some cases, very accelerated, or in other cases, just very drug out, right? So, you know, for some of us who don't have data collection ongoing right now, we're trying to pull data that we haven't analyzed before to get that out there. You know, it's going to be some time for us to actually get our data collections back up and running once we're able to see human subjects again. So, you know, you need to be realistic about your productivity when things like that happen. But also you need to communicate, right? So if you sense that somebody in your group is really stressed out, you need to figure out what's going on. A lot of times when I've seen this in other laboratories, there's something going on in which a student is stressed, either about an employment situation or a visa situation that might be causing them some problems. And so just check in with that person and see what's going on so that you don't have issues later on, right? I've just seen and heard so many stories in which people have not done that, and it's been a problem. Other things that you can do to address bias in publication reporting, always remember to report on your randomization techniques, your blinding, your sample size estimations, and your data handling. And these are things, oh, I am sorry, we have a special guest right now. Her name is Sarah. She clearly is reinforcing this, but she wants you to look for these hallmarks in the literature when you are building and reporting your studies. To reinforce what Melissa said, please use the Alcoa principles for managing your data quality, and maintain your data integrity using complete, consistent, enduring, and readily available sources. Generally, you should know the strengths and weaknesses of your teams and team members. Familiarize yourself with the expectations of your editors, your review boards, your funders, your institution, your boss. Know what they expect of you and when they expect it of you. Know your deadlines. know the necessary pieces you need for reporting, whether it be to a funding agency or you're, submitting a proposal for the first time. That's something that I've definitely seen junior PIs have issues with. They're not sure what is expected with them. And so they come up to these really massive and super stressful deadlines when they go to submit things. And also know the resources that you have available to you, right? And those resources can be administrative personnel, they can be submission portals, they can be any number of different things, okay? Know what those are, know who to contact, be kind to your administrative staff because they can save your butt, right? And so as we move into our breakout groups, I have a challenge. And so when we get into this breakout group, I want you and your team members to think about the things that I've talked about today and think about, 2021 is coming up, okay? So what can you do personally or your lab could do to improve your relationship with publication reporting and biases, right? So I want you to brainstorm 2 to 3 resolutions and strategies that you would be willing to share. So the group reporter would be willing to share when we come back to the group. So I will try to get this. I'm going to copy and paste this prompt in the chat. And I believe, Suzanne, are we ready for the Zoom rooms?
Yes. Dawn, are you going to open up the breakout rooms?
I'll open them right now.
All right, welcome back, everyone. So sorry to pull you back a little bit early, but we want to try to finish on time today. Does anybody from any of the groups, there are 10 groups, does anybody have something that they really want to share from their breakout for their strategies to improve their relationship with publication and reporting for 2021? Don't all go at once.
Hi, this is Kelly from Group 10. I'll go ahead and sort of, we basically talked about, we think as a group that the collective exposure to these sort of workshops is allowing us to, when we go back in the lab, and, a lot of it has been very different this year in regards to, everything's very Zoom meeting-like and stuff. So, so having, lab group meetings is not what it was, but it is allowing us to go back in the lab and discuss with our peers the workshops that we're all being exposed to lately. And we feel like there's been a huge increase in the availability and of these workshops, and that's actually allowing us to communicate more at the interpersonal level when we actually see people in the lab. Oh, I attended this sort of workshop. Hey, I learned this, or, vice versa. So I think that this exposure to our peers and to our collaborators and attending meetings that are creating this awareness is actually, as I can compare this time to last year, and I hadn't attended as many workshops as I have this year. And the exposure has always been there, but the fact that we have now some time to devote to it, I feel like this year has been extremely helpful. And going with that mentality into next year would be sort of a New Year's resolution.
That's awesome. That's really awesome. Working on interpersonal communication. That is the first time that anybody has brought that up here. So thank you. That was really great.
Thank you.
We have somebody in the chat that totally agrees.
Mm-hmm.
It's much easier to attend meetings via video conference.
What else? What else did we find out in the Zoom rooms?
Hi, I'm Michael from Group 3. I just wanted to kind of, like, towards the end of our chat, that same sentiment was brought up, that for a lot of us, as we've gone through a lot of our training, we've been inundated with things like ethics courses and workshops, but not necessarily as strong of a focus on something like rigor and reproducibility and how to address a lot of these things, like our relationship with our data and others in the lab as well. So I think that that's one of the huge benefits of something like this. I think from another resolution standpoint was we were discussing things like how a lot of people, even while they may otherwise be a great scientist, they may feel that they've kind of validated and verified things so extensively to themselves that ego might get into the way a little bit. And that can make it where someone is maybe trusting but trying to verify, the act of verifying is taken as a sign of lack of trust. And that one of the big resolutions that stems from that is to work on these interpersonal relationships and make sure people understand that it's not like you're not being singled out, you're not being called out. We're just trying to verify that everyone is doing the best science that we can, and that we're going to move forward with a reliable result and a reliable publication to come from it.
Awesome. Yeah, that's a really great point, is that some people can take this very personally when they're asked to verify. And we're not trying to be personal with it, right? We're just trying to be the best scientists that we can. So absolutely fantastic. Thank you. I think we have time for one more group. Suzanne?
Well, I was just going to read something and I thought was really good that I got from Muhammad Saad Shamim. I think that this is so practical and so simple, but sharing a first draft as soon as it's ready with the whole lab and involving everyone in the process would be good. I think that isn't just an excellent, simple thing that can really make a big difference. Get lots of eyes on it early.
Okay, hi, this is Rupali. I'm from group 6. I want to share that because of this COVID-19, we couldn't meet to our lab mates that much frequently. So if you are in the lab, you know what others are doing and you can talk and get feedback from each other. But because we are not meeting personally that much, and we don't have much idea what others are doing, because only just when they report the weekly things. So what should each member from the lab should do? They should take a turn and present their work from what was their objective and what they have done so far, and get the feedback what they want to do again, because when as the work progresses, you may not know what exactly your colleagues are doing, but the feedback from the colleagues is also important. So that way you have to have those presentations, not thorough presentations, but at least have goals and other things, the complete objective of the work and what work you have done so far. And also you should have the you should share your resources codes and data on timely basis. It is more important now than before, because before you could just meet and ask for the person, hey, can you share that code, or can you share that data? So it is now much important that you should share it on timely basis, update it regularly.
Absolutely. Absolutely. Thank you.
Mm-hmm.
So Shannon,
Suzanne?
Yeah, I was going to say, Shannon, why don't you go ahead and add the post-assessment survey link to the chat?
And thanks everybody for staying on today. I know it's a long day, very long day of COVID Zoom. So.
Okay, so that's the link is in the chat. So once you have submitted that, you're free to go. I do want to thank you all again today for your patience, and thank again our speakers. We appreciate all of you. Hope you all have good and safe holidays.